and welcome to the third CAPITALISE training workshop, Introduction to Field Trial Design. I'm delighted to say we have three experts in the field to speak to you today. We're going to start off with Dr Tally Wright, who is a group leader in quantitative genetics at NIAB in Cambridge. He has expertise in statistical genetics, experimental design and data analysis, and he's going to talk to you today about the basics of field trial design and analysis. Following that, we have Dr John Ferguson from the University of Essex and also the University of Cambridge, and he's a member of the CAPITALISE project involved in designing and implementing approaches to photosynthesis related traits and in key crops. He's going to highlight the steps involved in designing a field trial. Following that, we'll have a short break for about 10 minutes, and then we'll have our final speaker, which is Professor Johnathon Napier. He's based in Rothamsted, and he's renowned for having carried out some of the first and most significant GM field trials in the UK, looking at biosynthesis of omega-3 fish oils in camellia. And he's going to talk to us about his insights in translating from research to commercialization. But before we begin, we're quite interested to see what experience you have in field trial design. So I'm going to open a poll, and it's just for fun. Just click on what your experience is. So, have you conducted field trials before? Are you planning to conduct field trials, or are you just interested in learning more about field trial design? And as you can see, the majority are interested in learning more about field trial design, which is great. There's a significant amount of conducted field trials and are planning. Also interesting for us is where you work. Are you academia based? Are you industry? Are you both or other? And the results, as you can see, there's a majority in academia. There's also significant in industry, so that's nice for us to see. So without further ado, I'm going to hand you over to Tally to present for us.
Hi everybody, my name's Tally Wright. Thanks for the introduction, Louisa. So I'm a researcher based at NIAB in Cambridge. Today I'm going to be giving a fairly practical guide to utilising relevant experimental design and some trial analysis, which will hopefully be useful in your future experiments. I can see some of you have done field trials already from the poll, but I hope this is still relevant and useful information. I won't go into too depth in terms of the stats side of things, but I'm hoping what I cover is fairly relevant. So I'll be giving a brief introduction to the quantitative genetics group at NIAB, going into the importance of experimental design and why we should keep to certain principles, looking at trial design, and then going into two methods of analysis, which is the mixed model and spatial modeling methods, and then just to summarize some pitfalls to avoid when running these experiments and running the analysis downstream. So the quantitative genetics group at NIAB is really diverse in what we do. We do a lot of sort of different statistical genetics approaches, a lot of work using R, the sort of standard statistical software that most people use nowadays. We do a lot of downstream genetic analysis, so QTL mapping, genomic prediction, but all of this is based on getting this bit right. And this is the bit I'll be talking about today, which is the statistical analysis. So trial design, trial analysis, and trait modeling. So the more accurate we are here, the better these two approaches are downstream. So it's really about getting this side of things right. The other side of what we do, and this is, again, this isn't what I'll be talking about today. I won't be talking about the statistical genetics side. It is looking at how we can improve modern bread wheat in particular, using ancestral varieties, progenitor species that harbour sort of beneficial diversity in terms of environmental resistance, drought tolerance, photosynthetic productivity. So this wheat, we try and identify interesting donors that could be used to improve modern bread wheat, and then we try and have a lot of integration pathways to get this diversity into modern bread wheat. So for instance, backcross assisted marker selection. Another aspect of what we do is training. So we've been teaching historically at NIAB and a very popular quantitative genetics training program. So it's been run for 13 years. We've had to cancel the last couple of years due to COVID, and at the moment we're having a review on how to teach the course going forward. So I'll keep you updated on that at the end. So now we get on to the actual presentation. So experimental design and why is it important? So Ronald Fisher was a famous statistician that I'm sure some of you have heard of. A lot of his experimental designs we're still using today. So he famously said to call in the statistician after the experiment is done may be no more than asking them to perform a post-mortem examination. They may be able to say what the experiment died of. And I think this is a really relevant point, and it's definitely something I've been guilty of when I first started research, is designing experiments, collecting the data, and then thinking about the stats analysis. And at that point, typically you may have done something wrong, and it's really tricky to then go back and make the changes, and you end up not being able to do the analysis that you wanted to do. And it's something that we still observe frequently. So to get around this and avoid this happening, we need to cover these bases of experimental design with every experiment that we do. So these bases are replication, randomization, and blocking. And I'm sure a lot of these aren't new terms to you, but it's good to go over them here. I'll particularly focus on blocking. So replication, I'm sure most of you are aware of the importance of replication. There's some statistical jargon here. So treatments are the things we want to test in the experiment, and we can have different levels of treatment. For instance, we can have different genotypes. And then experimental unit is the smallest division of experimental material in our design. So that could be plots or pots. So a lot of this is kind of based around field trial design, but this is still really relevant to glasshouse bench experiments and growth chamber experiments as well. So when we have our plots or pots in the glasshouse or the field, the response will vary between the experimental units. There will be a different response in the trait due to that plot or pot in terms of where it is. No matter how uniform the environment, there will be a difference in response. So if we only have one replicate of a treatment in that pot, we have no way of telling where the variation in the trait is coming from. Is it due to the treatment effect, or is it due to that plot effect? So that's why we have replication. From a statistical standpoint, we need at least 2 observations to calculate a variance, and we need a source of error for significance testing. So randomization, again, I'm sure this isn't a new term for most of us. So fertility fluctuations, which can be anything in the environment, such as soil moisture or an outside window by a glasshouse bench, have a directional gradient influence across our experimental area. So this is a glasshouse bench. Typically, glasshouse benches are really light limited. Even if we have supplementary lighting, the plants grown on the benches are always light stressed. So even if you have an outside window next to one side of the bench, you can have a really dramatic effect across the bench. So in this case here, this is what we wouldn't want to do. We wouldn't want to have genotypes non-randomized. We have replicates, but non-randomized genotypes. So genotype 6 is going to have a advantage over the other genotypes as it's next by the window. The solution to get around this would be to completely randomize the design, which gives us a completely randomized design, which is probably the most simple acceptable design for analysis. And here we've randomized our genotypes and spread out that effect of the window gradient. Still, this can give us some problems, and this is why we get on to the next step. If you have a look, genotype 5 is still on the first 3 rows of the design. It's still going to have an advantage across the experimental area compared to the other genotypes. And this is something that we observe frequently. If we just use randomization, it often happens that by chance, all your controls end up on the perfect side of the field, and you end up skewing your experiment. The way to get around this is to use blocking. So we have one rep from each genotype in one of these 6 blocks. So we have 6 genotypes and 6 replicates, one each in each block. And that means that because we're blocking away from this directional gradient here, we spread out the effect of the window by having a variety in each block. And if we use this design, it's called a randomized complete block design, and you'll come across this a lot in research. So it's a randomized complete block design. One rep of each genotype is in every block. It's balanced. So that's a brief introduction to the sort of benefits of experimental design. So now I'll talk about trial design and analysis. So randomized complete block designs are effective when you have a small amount of genotypes and the trial conditions are fairly uniform. So in this case here, we have a block size of 6, which is a good block size. So we can use a randomized complete block size, randomized complete block design, because we only have 6 genotypes. And the benefit of doing that, if we look at this made up data here of this trait effect, we can see that the genotypic effect is present, but it's quite variable. And if we were just to do a basic ANOVA on this data, you see that there is a significant genotypic effect, but the leftover residuals is very high. There's a lot of unexplained noise in the data. If we were to include block in the analysis, we have a 2-way ANOVA here, we can see that we've really reduced the leftover error in the data by taking it into account in the block effect, which is very significant, and also our precision on our genotype estimate has increased and we've got a more significant value. So therefore, having the blocks taken into account in the analysis gives us better precision in estimating the genotype effect, and that's why the randomized complete block design is effective. So randomized complete block designs were very popular, and that's because they can be designed by hand and also analyzed by hand as well. I mean, this is just an example of how to do one using Excel. You'd have every replicate of your from each of your genotypes in one block. You'd have a different randomization column for each block, randomize using a general function in Excel, and then sort that column for each block on that randomization. And that's how simple it is to do, and that's perfectly acceptable. For more complicated designs, and I'll talk about this going forward, we can use different R packages. These are 2 R packages here, which are useful for more complicated designs, but these can also do randomized complete block designs too. I typically use this one blocks design. In the PDF, which I think you can download from the meeting software, all of these links to every package I mentioned and all the resources and references are downloadable in a PDF, so they're all there. So when randomized complete block designs are not useful is where we have a large amount of varieties, and that's because to include every variety in a block, we end up with a very large block, which means plots in that block, or plots are very far apart, and we lose the precision in estimating the actual fertility effect due to having a very large block size. You'd have neighboring plots of different blocks closer to each other than plots within the same block. So to increase our resolution in the correction for field fertility effects, we use an incomplete block design. So an incomplete block, we don't have a rep from every genotype in a block. So here we have an incomplete block size of 12, and so there's 12 pots in that block, and different. They're all from different varieties, but not every replicate. We have 72 varieties here, or 72 replicates, and they're not all represented in this block of 12 plots. So this is called a resolvable incomplete block design, because we actually have these complete replicates here. So there's a lot of confusing terminology here. Sometimes these are called reps, which is what I tend to call them, or complete blocks. But it's resolvable because if we ignore the incomplete block design, we actually have an RCBD design. This is also called an alpha design or an alpha lattice design, but it's basically all a form of incomplete blocking. So I just like to call them an incomplete block design. So now that we've got our sort of experimental design or trial design in our head, we can start thinking about the model that we're going to be using to analyze the data. So this is a sort of standard linear model. And I think it's a nice way to actually visualize what you'll be doing in the analysis, the model that you'll be fitting. all this model's really saying is we can predict the individual plot yield, or estimate the individual plot yield of a plot anywhere in our experiment using this equation. We take our experimental mean plus the variety effect for whatever variety is in that plot, plus the block effect if we include a block, so where that where that variety is in a certain block indexed by J, and I indexes which variety it is, and then plus the leftover error for that plot. So if we add all of these together, like we have in this example here, we end up with the individual raw data point for that plot yield. And it's just a nice way of working the model backwards. Where things get more complicated is where you have two-dimensional fertility effects, and that's often the case. Fertility effects are rarely easily detectable. You don't often know where the fertility effect is going to be. It's not like a window by the glasshouse bench. So you can't always organize your blocking moving away from that directional gradient you're expecting, which is what you want to do. In some cases, it's better to cover for any event and use two-dimensional blocking. So a really good example, and a very popular example, is a row and column design. So here we've lost our incomplete blocks on the last slide, but have a different form of incomplete blocks, which are just row blocks. So we have a block size of 6 and column blocks, a block size of 12, again in each replicate. So it's still a resolvable incomplete block design. And then in our model, how we'd include that, we'd have a term for complete block or rep, which is B there, indexed by J, plus a term for row, but it's important to note that we're nesting that within the overall block effect, and that's because we're interested in testing the effect of row 1 in this full replicate here, not row 1 across the entire trial. So it wants to be nested within there, and again column block nested within complete block. So creating an incomplete block design, you can't do it by hand. It's very difficult because there's a lot of things that need to be taken into consideration. Traditionally, the approach was to use or modify previous designed experiments from textbooks such as Cochrane and Cox, which is the classic one that everyone used to use. So actually, this approach is still used in many breeding programs and recommended list testing for different varieties nowadays. It can be a little bit wasteful because to get your numbers to a certain a certain number of varieties included in the trial, you have to sometimes increase what you're entering into the trial to match the previously designed experiments. And also it can be quite restrictive. One of the issues I constantly have is that the experimental area changes so much. You're constantly having to work with different block sizes and different rep sizes, so it's really hard to try and fit these into a previously designed experiment. The solution is to use computer software, that should be computer software, to create and optimize the design. And then these packages in R are great for that. I typically use Blocks Design. And these are why it's important to use computer software, because it gets very complicated. There's a lot of underlying principles in the incomplete block designs that you should consider. Multiple levels of randomization, grouping treatments into different sets for different blocks, allocating those different sets to the blocks, and then within block layout, all of those levels of randomization. No repeated treatments within each block. It's better to keep an even distribution as much as possible. The pairwise occurrence of 2 genotypes across multiple blocks should be minimized, and that's really important because it might seem like an overcomplication, but if you have a tall genotype and a short genotype, you don't want them always to be next to each other in the trial, because the tall genotype would always shadow the small genotype, and you'd end up with a confounding effect due to the height difference. So it's good to spread out the pairwise occurrence of varieties, and most of the software will do this for you. And also it does really clever things like if overlapping occurrences of variety A and variety B and A and C occur, then there's less requirement to compare B and C. So it does a lot of complicated things like that, and it's better to get the computers to work all of this out. And how it does it is most of the software will compute what's called an efficiency metric, and this is a comparison of the efficiency of an incomplete block design to a randomized complete block design. It will go through a lot of iterations to find the design with the highest efficiency. So these are some more logistical considerations that I've put in, and And it's actually, I think it's really important to think about these, because you can do a perfect design on the computer and you think it makes so much sense to you, and then you hand it over to the farm team or whoever's drilling the trial, and there'll be problems that you hadn't thought about. So when it looks good on the computer, you need to think about actual plot size and shape. Ideally, square plots are better, but we really rarely have square plots. They're normally rectangular, so you need to think about your block sizes. You want to keep your block size relative, sorry, your block shape relatively square too. Tram lines and other physical separators really give me a headache when designing experiments. And this is one that I designed last summer, and it was analysed this summer in the field. So it's absolutely massive. It's 2,500 plots, and there's a massive tram line going up the middle of the trial. So I needed to take that into account in the design of the experiment. What I didn't take into account, I probably wish I had done now, is that there's actually quite a large gap left between nursery lid drilling. So you can see that there, and I wish I'd taken that into account when forming my blocks. So that's something to think about. It's important to go to the trial and have a look. And we don't always have like a perfect drone image of the experiment like we have here, where I can clearly see what I wish I'd done things better. But in retrospect, it's important to go to the trial and have a look, because you might actually realise that once you're there, the whole experiment's on the side of a hill, and then it's quite useful for expecting what the spatial gradients are going to be like. So block size, it's variable depending on what you're working with, if it's pots or in a grow chamber or field, but 5 to 15 is what I've used in the past. Edge effects. So one really common mistake that we've heard quite regularly, and it's things that I've done before, is that people assume, for instance, in the glass house, there's going to be no gradient because it's in the glass house, everything's fairly controlled. There always is a really large gradient, and we found it's better to have a row of plants around the edge of the glasshouse bench to remove that edge effect. In field trials, there's always discard and actual commercial varieties growing around the edge of the trial to reduce the edge effect too. So communication with the farm team and the trials manager is important. Make sure they know where plot 1 is in your trial and when they're drilling it, so that it's not drilled back to front. And yeah, those are some key things to look out for. So there's a lot of designs that I haven't really gone into. I just focus on sort of the incomplete block design, but there's more you can use. You can use an unreplicated design. So that sounds like it's a bad idea, but often in breeding programs and some of the pre-breeding work we do, we don't always have enough seed for replicated plots. The way to get around that is, of course, still to use randomization, but also to have control checks spread out through the trial very regularly. So 1 in every 5 plots, around 20% of the trial should be control checks. And this means that although we can't use the unreplicated ones to make corrections, we can still use the check varieties and use that to make corrections on the unreplicated varieties for fertility effects. So it's better to use multiple checks, because one check might fail in the experiment, and then you're in trouble if that happens. Or a partially replicated design, you might have some replication available for some lines. So again, you'd have a proportion of the trial replicated and use those to correct the rest of the trial. So one thing I would recommend here is to use super blocking. So this is a way of making sure that you're spreading your checks evenly out across the trial, and all of your checks don't end up in one corner of the field. And don't forget to randomize. So that's the trial design aspects of the talk. So now we can think about preparing the data for analysis and things to do beforehand. So a lot of my time is actually spent on data QC. I think I probably spend about 90% of my time on data QC and 10% of my time on analysis, which isn't ideal, but it's unavoidable. It takes a long time, and especially if multiple people have worked on a field trial, there's a lot of data QC, putting the data together that needs to be done. So it's a step that's really important. One of the first steps is to check for extreme values and mistakes in data entry. They're always there, you just need to find them. Converting the data into an R-safe format. So for my analysis, I use R, so this is very R-tailored. I use read.csv, which I think is a safe way to enter data in. Outliers is something that's always tricky, and I'm sure we've always come across these problems. if you've got field notes to go along with the data, you can immediately check any outliers to see if there was a drilling issue or something. And yeah, be careful with removal. Normally you need a fairly good reason to take them out. Plot data as a histogram. So that's the key thing, is to plot your data before you run your stats, and just get a feel for the data. Identify any outliers, identify any mistakes, look at variance between lines, and then check format of data columns and missing data entries. So if you've got missing data, make sure it's entered it how you want it for whatever software you're using. And this is something that I say is something to keep an eye out for. If you've got a numeric block number as your block information in a data frame, make sure it's been treated as a categorical factor, not a numeric factor. Sorry, not a numeric variable. So this is something that I've started doing over the last couple of years, and it's really useful in field trial analysis. I think it's one of my key steps now, so I've added it in here. It's using a package called DesPlot. And what you're basically doing is you're plotting your raw data across a grid of your field trial. So if you have row and column information of the field trial, which you should have, even if it's not a row and column design, it should still be a grid, so you can get that row and column information. Then you can actually plot the trial, how it was laid out in the field, and you can immediately see any spatial patterns. So this is a really strong spatial pattern here for grain yield. This is one of the more extreme ones that I've seen from a trial this summer. So you can see this is a completely randomized trial, but we have this group here of low yielding plots, and that's how strong a spatial effect can be in the field. And then a very high yielding area over here. So this is what we need to take into account in the analysis, because if it's not taken into account, we're going to have a very large error term, and our precision in estimating genotypic effects won't be very accurate. So this is why we need to take into account these field fertility effects. And then going into the analysis, you want to make sure that you're clearing up this noise and the residuals left over, and I'll talk about that in the next couple of slides. So modelling, we're on to the sort of stat side now. So there's two sort of main approaches which are typically used, so mixed linear models and spatial models. So mixed linear models are the most common used method for analyses in crop science. They're used for analysis of incomplete block designs, unbalanced experiments, G times Z investigations, genomic selection prediction, genome-wide association, GWAS, and many more. So it's a really useful skill to pick up, but it can be really complicated to get into, and there's a lot of fairly confusing literature out there, but there are some good papers. I think one of them I've included in the references, so it's worth a read. And then so software for doing this, a lot of my time I use R with LNE4, which is the main sort of linear mixed effects model package that people usually use. Some as another alternative, or that there's paid software where you have to pay to use. ASReml is very popular. If your organization has a license for that, it's good to make use of it, and GenStat as well, which is outside of R. So the mixed model. So if we go back to our very simple model here, we can predict or estimate an individual plot yield in the trial by adding our variety effect indexed by I for a certain variety, whatever is in that plot, plus the experimental mean, plus the residual error, and that will give us that plot yield. So this is what we're ultimately doing when fitting a linear model. It's a simple model. We can add in a block effect by adding this term here, so just adding it in, and J is indexed by whichever block that plot is in. And then if we're considering this model as a mixed model, what happens is these factors are either fixed factors, so in this case the variety effect or genotype effect is fixed, but our block factor is random, and our residual error is always treated as random as well. So the block effect and error term are considered random, and the difference is these random effects have a mean of 0 as they deviate from the experimental mean. They also have an unknown variance component, and they are considered to be normally distributed. The normal assumptions of a standard linear model apply, as with a mixed model, so the independence of data points and homogeneity of variance, and the normality of distribution of residuals. So why? So what's the difference between fixed or random, and why would you have varieties as fixed and random? This is a bit of a grey area, and there's lots of discussion and debate over what should be treated as fixed or random factors. So this is a general rule of thumb that I sort of pulled together from advice from different people, and I think it's fairly useful, but not always completely accurate. So fixed factors do not come from random samples, and our experimental variables we are interested in testing. So they are unknown effects that we want to estimate, such as genotype, nitrogen application, and fertilizer response. So these are the factors we're sort of interested in working out what their effect is within this experiment. Random factors come from random samples from a larger population and/or are things we are generally not interested in. So this can be things like block, sampling date, a scorer bias if you've got multiple people scoring your trials, or year effects, for instance. There are some exemptions to this rule, and a good example is genotypes that I'll talk about in one of the later slides. So why use random factors and why use the mixed model? What's the advantage? With fixed factors, your results are relevant to the discrete factors that you've tested. But with random factors, you can make predictions of the population the sample is being drawn from. For each random factor in the model, we estimate a variance component, which is used to estimate the importance of the random factor and the variability of the response variable. For fixed factors, we estimate their significance in the standard sort of F-test approaches that we'd expect using an ANOVA or a standard linear model. Random effects have this advantage, which is called shrinkage or pooling. This is where they're estimated with this shrinkage, and that means that predictions of treatment levels that have fewer replicates are influenced by those that have a greater number of replicates, and that means we can improve our precision of genotypes that have fewer replicates in the trial using the information from those that have greater replicates. So where we have varying amount of replicates, this is a really useful approach to use. In terms of the incomplete blocks, if they're in the experiment, why treat them as random? It's because of this recovery of interblock information in the modeling. If they were fixed, we'd just be running intrablock analysis, and this is where we account for the block effect and then work out how much additive variation is left over for estimating the genotype effect. So that leftover variation is explained by the intrablock variation. If we treat them as random, we move to this interblock analysis where we use the information in block means that is linked to the interblock variation caused by which genotypes are within each block to improve our estimate of treatment effects. We're utilizing that information, that extra information, and the method for doing this would be to treat incomplete blocks as random using the software such as LME4 in R. So blues and bluts, if you get into crop science and field trials experiments, these are terms that you're gonna hear a lot. In some cases, as I mentioned, it's better to treat genotype as random, for instance, where you have different levels of replication. The difference in treating genotype as random or fixed comes down to estimations of their means. So we estimate blues in this case, which are best linear unbiased estimates, where genotypes are fixed. So these are estimated in the sort of standard least mean square way that we'd expect from a linear mixed model. They're just our experimental mean plus the variety effect, and that's replicated over the different replicates across the trial, and that will give you a blue for a certain variety indexed by I. However, if we move to working with our BLUPs, our best linear unbiased predictions, then we'd have genotype as random, and here what we've done is we'll estimate the equivalent to a blue, and then times that, multiply that by what's called a shrinkage factor. So this shrinkage factor comes from a function of treatment effects variance divided by error variance, which is sort of equivalent to heritability and is influenced by replication number. A little bit clearer way to think about it is more replication would equal less shrinkage, as we'd expect. Thinking about the advantages of having it as more replication, and then more extreme values equal a greater shrinkage to the mean. So treating genotype as random has a shrinkage effect. So thinking about this more practically, an example of where to use BLUPs, as I mentioned, if the replication is uneven across different genotypes we're testing, the shrinkage factor would vary across variety, and that means we'd get a better accuracy and precision of the low replicated varieties than if we were to use no BLUPs. An example of where to use blues, so if we're interested in testing a more simple hypothesis, such as is the applied herbicide better for yield, or if we just want to compare mean environmental performance, mean genotype performance within a given environment, and replication is fairly even, then it's easier to use blues, and they're a little bit more robust in what you can do with them afterwards as well. So yeah, there's probably not time to give a complete workthrough of this, but this is a sort of thing that I would do in R when coming to do a mixed model analysis, and I hope this is useful. So let's say we have an incomplete block design here, a resolvable incomplete block design. So we want to compare mean performance between genotypes within a single environment. Then our model would be this. We'd have our overall mean plus the variety effect plus the replicate effect plus block nested within rep, those being complete replicates blocks, and these being incomplete blocks, plus the residual effect. So that's the model we want to test. So in R, I'd use the package LME4 for doing this, and this would be the syntax for how you test it. So you have your response variable, genotype as fixed, as you would in a standard linear model, plus the random term. So this will test the effect of rep, and then it will also test the effect of block nested within rep. And with random terms, they're in brackets with a 1 and a pipe starting at the start of the term. Example.data is our data frame, and we're testing with rem all equals true, which is restricted estimated maximum likelihood, which is the preferred method for testing mixed models. So we then go through a model reduction stage. So this is where we drop variables that have negligible effects in the model. So I don't have time to go in this too much today, but we're basically, as we would in a multiple linear regression, want to reduce the model to the components that are have a fair effect in the model. We then can check the model diagnostics, so making sure that we've met all the assumptions that we're meant to, and then explore the model results and make inferences, look at the significance of genotype, how good the heritability is for a certain trait of interest. You can do this using packages such as LMER test, look at the genetic effects, and then extract error estimate predicted means. If genotype is fixed, we're getting out our blues, and rank and compare variety performance. So we can do that using EM means, which is a package that extracts the blues from the model. So that's an example worked through for an LME4 approach. One thing I say, expanding on that model diagnostic step. So actually, I found this package when preparing for this talk. I used to use a different one, but this one looks quite helpful. It gives you quite a lot of information, and it also gives you what you should be expecting in each plot as well, which is always helpful. So it's always good to have a look at the model diagnostics. I mean, typically you know beforehand if there are going to be some issues in the data. This is from one of the trials that I've actually shown in this presentation. The two graphs that I always have a look at to make sure that they're in line with what I expect is the normality of residuals and also the homogeneity of variance. So this plot's really useful because what you'd expect to see is a random distribution of the residuals across the fitted values, and there shouldn't be any correlation between the residuals and the fitted values. They shouldn't get larger as the fitted values get larger. If that's the case, you've missed a trend in your data, and that's normally something that's been missed in the experiment. There's a confounding effect that you kind of need to go back and work out what's causing it. One example of that is from this field trial here. So this is actually the one that I showed right at the start that had this 2,500 plots. So this is from this summer. There's quite a large gradient that you can actually see in the raw trait data here. There's a lot more extreme values on the right-hand side than on the left-hand side. Where this has come from is a scorer bias, so that there was two people phenotyping the trial, both very experienced, and I think they did the first run together and then went their sort of separate ways and scored the experiment. And yeah, when I got the results back, I noticed that there was this trend in the data, and I asked them which side they scored, and the cutoff was about there. So we can take into account the scorer effect, and it's not a problem because they let me know that they scored half of it each. So it was something I was looking out for, and it's something that can be taken into account in the model, and we can correct the data for it. So once I've fitted a model, I always use this step which is to compare the raw trait data across the grid of the field. You can do this with a glasshouse bench. I know it's not normally perfectly square grid, but you can sort of estimate it, or a growth chamber, and then compare the residuals after the model fitting. So this is what's left over that wasn't accounted for in the model, and you can see that after fitting the model, we removed this spatial patterning, and it's more of a random variation in the noise that's left over. Yeah, so this was using a row and column design nested within Replica, which has cleared up the spatial variation quite a lot, and it's a much more even distribution. There are still some areas where it looks like there might still be something remaining. And one thing we can do is now move on to the second step of modeling if we wanted to, or you can start out by using this approach if you'd rather, and that's using spatial models. So traditionally, autoregressive models have been typically used for spatial analysis. These are available through software such as GenStat and ASReml. So I'm not going to talk about these today. I'll be talking about a free approach of spatial modeling that has become popular that uses two-dimensional penalized splines. So this approach is free, and it's become very popular in crop analysis. It's something to look out for. It's a really robust, quick way of analysing a field trial. So a penalized two-dimensional spline, if you imagine a one-dimensional spline as a curved line with a fitted trend, then we can have a two-dimensional spline as a sort of more of a bendy sheet. That's not a very mathematical way of explaining it, but I think that's a good way to visualize it. So you kind of fit in this bendy sheet the field fertility patterns of your experiment. So how will we implement this spatial model? So SPAX is an R package that runs this penalized spline ANOVA. It consists of these different smoothing components dependent on a single smoothing parameter each. And the model also takes random factors and fixed factors, which is great because you can include the other terms of your experimental design into the model, which is definitely appropriate if you do have another design there, and other factors that you want to include, such as covariates or other nuisance factors. You can output blues and blups. You can also output heritability, a generalized heritability estimate from the SPATS model. So it's quite useful for using. And that's how robust it is. But actually, in terms of the output you get from the model, this is the residuals left over from the row and column block analysis. You can compare the residuals here. There's a lot less visual variation left over in the field. It seems a lot more random. That might just be because the color scale is a little bit more forgiving, but it's something to look out for. So once you fit your SPATS model, you want to have a look at this and make sure there's nothing left over. You can see the raw data plot here. So this is what we were plotting originally, the fitted data trend there. And then this is the fitted spatial trend. And this is what we'd expect to see based on the raw data. So you can also see that if you look at the values here, you can see that they're on the same scale and magnitude as the residuals. So that shows that the fitted spatial trend is quite a strong one, and we can again get our genotypic blues out of SPATS. So this is quite a robust method for analysis, and that's why I think it's becoming very popular, because you kind of fit everything in one model, whereas in the linear mixed model approach, you'll have to compare lots of different models to come up with the best one to use. So in summary, some pitfalls to watch out for, to summarize the talk really. So no replication, no randomization or blocking. I hope I've communicated that across, that it's good to use replication, randomization and blocking. They're very important for when it comes to analyzing the data. And yeah, if you want to do the proper analysis, you've got to take those into account. Thinking of the analysis or consulting a statistician after data collection, it doesn't have to be a statistician. Anyone that's got experience, ask for help before doing the experiment. Don't just go out there guns blazing and take the data without really thinking about the analysis you're going to do. I'm sure we're all guilty of doing that at some time. So pseudo replication, this is an issue I haven't really had time to talk about today, but it's really about knowing your technical replicates from your biological replicates, or your measurement units from your experimental units. So think about what your smallest division of experimental units is. It's normally the plot and the pots, or the pot, and that's something you can read up on online. There's a lot of good resources and examples out there for identifying it, because it's not always that easy to identify. Check data that should be categorical is being treated. So this is a classic pitfall, is make sure your blocks and reps are actually being treated as factors. If you're using R for analysis or even GenStat, something to watch out for, to make sure that they're being tested how they should be tested in the model. Look out for a scorer effect. We're all human. It doesn't matter how experienced you are. If you've got multiple people scoring field trials or making phenotype errors, always write down who's taking the measurement, because it could be something that you can correct for at the data at the end of the experiment, but you can't make that correction if you haven't recorded who's done the measurement. So non-linear responses is another thing to look out for. It's not something that I've covered here, but a good book for that is Mixed Effects Models and Extensions in Ecology of R. That's definitely one that's worth looking up, because it's really helpful for general mixed model stuff. Data QC, you can't skip it, and always plot the data. That's the key thing before you start the analysis. So I've listed some useful references here that I've spoken about throughout the presentation, and added a couple more that I thought would be useful. Added links to the each R package I mentioned. And this is available through a download in the handout section of this talk software. So a big thank you to these two people. So Ian McKay developed the quantitative methods plant breeding course at NIAB when he was at NIAB, and a lot of this material I've spoken about today has been inspired from or taken from the course materials. So a big thank you to him, and also thank you to Keith Gardner, who was involved in developing the course material as well. Just a note on the quenching course. So it's traditionally been a 2-week course that runs at NIAB every year. So it used to run in person. COVID made that really tricky. We had to cancel, and we had to cancel the run this year due to COVID as well. We're currently having a sort of internal review about how to teach the course going forward. At the moment, we don't have a date of when it will be taught again, and we're just trying to finalize some ideas on that at the moment, but keep up to date on the NIAB website if you're interested in that. And thank you for listening today. Yeah, thank you to the CAPITALISE project organisers for inviting me to speak, and I'll be happy to take any questions.
Thank you, Tally. That was great. I can say I've done the course at NIAB, and it's a really comprehensive course, and it's a very good course if anyone's interested. We're happy to take questions now. If anybody has any questions, either raise your hand or put your questions in the question box. I have one here from Subhana. I'm going to try and unmute. Okay, would you like to go ahead and ask your question, Subhana?
Yeah, are you hearing me?
Yes.
Thank you, Lisa. Thank you for the chance. Tally, I have a quick questions. Like you mentioned that we can use the mixed model for incomplete design.
Yeah.
That doesn't mean that I can't use it for simple RCBD.
A simple complete block design.
Simple.
Yeah, if that's the sorry, if you mean if you can use it for a RCBD design, a randomized complete block design, I think it's still useful to use for that. I mean, people have used RCB, the block effect in RCBD designs as fixed, but if you're using it as random, if the block effect is really large, you can recover some of that interblock information, so it's useful to actually use it as fixed as well. Although it shouldn't make too much. If it's completely randomized, it won't make any difference to genotype prediction. So if it's completely replicated, it's a full randomized complete block design, it won't make any differences to the genotype mean prediction if block is treated as random or fixed in that experimental design.
So you suggest me to go to a linear model, right? If there is not much block difference.
I mean, to be honest, I tend to, if you have got an RCB design, I tend to just use a linear mixed model anyway, and treat block as random.
Yeah, block as a random. Thank you. Thanks for your suggestion.
No problem.
One of the things that I don't think you mentioned was in doing experiments in different environments. So is there a number of times you'd recommend repeating an experiment in different environments? Is there a magic number, or is it so usually as many as you possibly can, but in reality, is there a recommended number?
Yeah, that is a really good question, and that's something I haven't really spoken about today, is like G times E modeling, and this has all been focused on having an experiment in a single environment, and obviously it's ideal to have multiple environments and do a more sort of multi-environmental analysis. Thinking about if there's a golden number and how many environments you have, I think people always try and weigh up, is it better to have more replication in one environment or lots of different environments and fewer replication in those environments? And I think the second option is better, is to have as many different environments as you can with fewer replication in each of those environments. It's difficult though, because if there's a strong spatial gradient in one of those environments, it's going to skew the results. So it is a trade-off, but yeah, capturing different environments is probably the best way to go, to try and get as many as you can, I guess.
Yeah. I liked your DES plot. I hadn't seen them done before, and the fact you could, from your DES plots, you could see your score bias on those, which you always feel there's a bit of blame going on there, isn't there?
Yeah, the guys who I showed it to, they are very experienced, and I think they're a bit taken aback when but it's something that it always happens, even if you have, it's like, measuring, if you're doing work with Lycors or any type of machine, there's going to be a machine effect as well. So it's something to always think about in your design, is how can we take into our, when we're using different machines, different ways of analyzing data. It doesn't matter if it's a person or if it's a phenotyping tool, there is going to be an effect if you're using different tools. So it's something to think about, and how to randomize that across the experiment is tricky as well. It's not an easy thing to plan, but yeah, it's definitely worth looking out for in the analysis.
I've got another question here, hand up from Janina. Let's try and unmute you. Janania, can you hear me?
Hello? Can you
Yes,
hear me?
We can hear you.
Yes. Thank you very much, Tally, for your nice presentation. I have a question. Is it possible to use another distribution like Poisson with mixed model in R when you don't attend the presuppositions, normality, homogeneity?
Yeah, that is a good question. It's a tricky one to answer, and it really depends on who you speak to as well, because there's a lot of people who aren't as worried about normality in the residuals. I mean, a lot of these analysis methods are more robust to normality than I think people worry about. I mean, sometimes we have transformed data if there is an issue in the residuals. If you remember, it's the distribution of the residuals to look out for, not the distribution of the data. It's the residuals that have to be normally distributed. So yeah, sometimes a log transformation can make an improvement on that QQ plot, and if that's the case, and sometimes it's better to use that, or just transform the data, test the result, and then test the result with the data untransformed. And if it doesn't make much of a difference, then I think it's okay to go with the data that might have a slight distribution issue. But yeah, it's a tricky one to answer that. There are some ways to analyze, some more formal ways to analyze non-distributed data, and that book I recommended about methods in ecology, mixed models in ecology, is a good thing to have a look at, I think.
Okay, thank you very much.
You're welcome.
Thank you. I have another question from David Casper. Is there a way to take into effect seasonality effects with only one growing season?
That's a good question. Not that I can think of off the top of my head. Obviously, it's easier when you have multiple, because you've got the issue of having one replicate there really, and that's where you run into the replication issue. You need to have multiple observations, so multiple seasons, to make those sort of differences. But yeah, I mean, have a look at the literature. There might be something out there using some sort of inferral, but it's not something I've come across before, so I can't help on that.
Okay, Well, I think we're going to leave. Oh, hold on. There's one more. Let me see if I can. From Savannah. If I used mixed model for ANOVA, can I still use LM model for correlation stroke regression curves for RCBD design field experiment? Want to read that again?
Yes, please.
If I use mixed model for ANOVA, can I still use LM model for correlation or regression curves for RCBD design?
Yeah, I might have to follow up on that one because I'm not sure if I really understand the question. So feel free to drop me an email with that, and we can follow up on it.
Yeah. Okay. So I think we may be able to do some more questions at the end, depending on time. But I think now we have to move on to our second speaker. So thank you, Tally. That was great. I really enjoyed it. And we're going to move on to John Ferguson.
All right, so thank you very much, Louisa and Christina, for organising this workshop. That was a really good start from Tally, and I think what I will present will follow on quite nicely. So my name's John Ferguson. I work at the University of Essex, and I work with the CAPITALISE consortium, where my main responsibilities are related to developing and utilising high throughput phenotyping approaches to characterize natural variation in the capitalized species of interest. So I'm really interested in developing and using photophysiological phenomics pipelines that allow us to gather vast amounts of data relating to aspects of photosynthesis that are potentially limiting productivity. So to give you a more kind of broader context with the work we do at CAPITALISE, we're interested in 3 species. So we work with barley, tomato and maize. So these are 3 very important crops for European agriculture. And so for my part of the project in particular, I'm interested in using these kind of pipelines relating to measuring photosynthesis to screen natural variation within these 3 species. So for barley, we work with 320 natural accessions of wild barley. So this is closely related to cultivated barley, but it hasn't undergone kind of breeding bottlenecks, so it potentially still harbours interesting alleles that might boost photosynthesis and be related to productivity. Similarly, with tomato, we work with a backcross inbred line population. So this is cultivated barley, which has large genetic introgressions of wild Pinelli tomato. And then with maize, we work with a multiple advanced generation intercross population, or a MAGIC population. So this is a population where we focus on 320 recombinant inbred lines, which have been derived from 9 genetically distinct founder lines. So today, for my component of the workshop, I'll give a kind of couple of case studies on how we analyze data that we get from our field trials, and I'll specifically focus on 2 examples that are coming from this maize experiment here. So there'll be 3 components to what I talk about today. So first, I'm going to talk about designing an alpha lattice field experiment and show you exactly how we do that within CAPITALISE. So this is an experimental design that we use for the 3 species when we're surveying this natural variation in fields. And then I'll give 2 examples of how we analyze data that we generate from these experiments. So firstly, some flowering time data, and then some data that's related to photosynthesis. So in specific, a photoprotection mechanism called non-photochemical quenching. And again, both of these are coming from the maize experiment. So what I'll talk about now a little bit is kind of echoing a lot of what Tally has already talked about, but a kind of a bit more of a basic level. So I guess, what is an alpha lattice experiment? So these are experimental designs where you have incomplete sets of treatments within a block. So treatments for us are genotypes. And these are really ideal designs when you have large numbers of treatments. And as which is the case for us, we have sometimes as many as 320 genotypes. So these kind of designs are often employed in large experiments and in plant breeding. where you can reduce the number of treatments or genotypes that you have within a block. And this can be, as Tally was talking about earlier, this can be quite advantageous if space is constrained. For example, if you're working in a glass house and you have a specific space that you have to fill. And that's been the case for us in the first year of CAPITALISE, where we had to run our tomato trial in a glass house because we couldn't get out to Italy where we would normally work to do them because of COVID restrictions. And this can also be important if your block means something in terms of an environmental or a biotic treatment. And kind of echoing a little bit what already talked about, the main sort of advantages of these designs are, the main sort of advantages of these tools when you account for the block effect nested within your replicates. And because we have these blocks, we're afforded extra precision that is completely cost and risk free. As with any design that you might work with, there's always disadvantages. And that's also the case with alpha lattice experimental designs. And for any particular design you might have, you have a fixed number of treatments or genotypes. In our case, you have to have a certain amount of genotypes for a design to be resolvable, any particular design to be resolvable. And then when you're modeling your data, we're accounting for block effect nested within replicate. And that can be a problem because our blocks are incomplete. So, and this is particularly the case if you're using this design with a small number of treatments or a small number of genotypes. And then because of that, it could be feasible that your block could be saturated with predominantly low yielding genotypes, for example. And in that instance, that low yield might be erroneously accounted as a block effect when it's actually a treatment or genotype effect. So this cartoon here kind of illustrates what a simple alpha lattice experimental design might look like. And in this particular example, we have two replications. So we have the same number of treatments or genotypes within them, but in a different configuration within and between the blocks. And within each replication, there are 4 blocks. And each block has 4 treatments or genotypes. So you can imagine each square here is a plot of a genotype. And that plot can be configured in any which way you require. But in our case with maize, we have 4 rows of plants with 20 plants in each row. So you can kind of define this specific example as having 32 treatments, or T being 32, a block size of 4 because each block has 4 plots, 2 replications, 4 blocks within each rep, or S, and a total blocks of 8. So the important thing to remember with these kind of lattice designs is the number of blocks per rep that you have multiplied by the number of reps must be equal to the total number of blocks you have. So S times R must equal B. So with all things being equal, and if you have no confounding variables that you wish to account for, and Tally has sort of gone over this in a bit more detail, you can analyze data that comes off of an experiment like this with the following kind of basic linear mixed model, where you're saying your response variable is defined as a function of the average, the treatment effect, the replicate effect, the block nested within the replicate effect, and any random error that there might be. So for our work with CAPITALISE, and Tally has already talked about this specific software example, we use the software package Agricole, which is executable within the free R software to design our alpha lattice experiments. Other packages are available, and I think Tally's reference in the handout might link to some of these that could be of interest. So now I'll switch over to my R software environment and show you how within CAPITALISE we use this package to design an alpha lattice experiment for our maize trials. So I'm obviously conscious of the fact that not everyone is familiar with R. So I'm going to try and run through this in a way that it should be understandable if you're not, or if you're just getting used to R, and perhaps if you're very experienced in R, this might be quite slow for you. In either instance, you're more than welcome to email me for the code and the data, which I'm happy to share, or if you have any questions, and my email address is on the poster, I think, for this workshop, but also at the top there. So, so I've pre-written this script and annotated it so you can see what we're doing, but I'll kind of describe as we go through exactly what we're doing. So the first thing we want to do is load up the packages that we need to design this experiment. So that's agricolae and a package called numbers. So then we're going to set our working directory to where we have a CSV file which contains our germplasm information that we need in order to generate the design. So I will read that in using this function read CSV, and I will call that object maize germplasm. So we can look at the top of that. So you can see that's in our environment here. And I'll look at the top of that now, the top of that file, just the top 20 lines to see what it looks like. So here you can see we have a data frame that has 3 columns, accession, name, and type. So accession is kind of a code that refers to each particular genotype. This is the name of the genotype, and this is the type of genotype that it is. So you'll see the first 9 are founder genotypes. So these are the parents of the recombinant inbred line population, and everything subsequently is designated as a real. So agricolae has numerous functions that start with design dot. So if I hit tab now, I can see some of the different options there are, and you'll see there's lots of different options for generating different experimental designs. So there's randomized complete block design here that Tally has talked about, lattice design, augmented design, etc. The one we need is design alpha, and you can use this function here to get some information, some help about it, and exactly how to use it. And you can see to design an alpha lattice experiment, you need to have some important argument, you need to input some information. You need to say, you need to define your treatments, or in our case, genotypes. You need to say how big you want your blocks to be, and how many replications you want. So to decide how big we want our blocks to be, we need to know how many accessions we have. So we can do that by using this function length and refer to the accession column within our maize germplasm file. So we have 320, which is what I was talking about, which is what I said earlier. So then we can use the divisors function to calculate potential size of the block. So these are all divisors of 320. Within CAPITALISE, for our barley and our maize, we've used a block size of 8. So we can go through now and generate our alpha lattice experimental design. So what I'm doing here is creating a new object called alpha lattice. I'm using the design alpha function. The treatments, I'm referring to the maize germplasm data frame and accession. So that's just pulling out all of these. And I want the block size to be 8, and I want 2 replicates. So I can go ahead and run that now. And then you can see some statistics to do with your experimental design. So we have 320 treatments, our block size is 8, and we have 40 blocks. And this is our efficiency factor, which Tally was talking about earlier. So, so you can see within this object, you have several different lists. You have the characteristics of the experiment, some statistics, sketches. So these are basically maps. So it produces a map of rep 1 and rep 2, and it produces a field book. So we can have a look at these maps now. So these are basically looking at the layout of the different replicates. So I can say view alpha lattice sketch rep 1, and that will come up here. So here you can see the rows are essentially your blocks, and these columns are the columns of the field trial. So I can also see the rep 2 one, and I can compare them. So this is rep 1 and rep 2, and you can see the genotypes are random and the blocks are random within the reps. So the other thing I mentioned that is created is a field book. So I can view that by running this. So here, each plot is assigned an identifier. So this first plot is 101, goes down sequentially, and it's given coordinates. So this is the column that plot is in, this is the block that's in, and that's the accession that's in that block. And it also tells you which replication it's in. So then I just have a permanent record. I can write this field book out, and I can decide the name of it here. So I'll do that now. So then if I go to my directory, which we referred to earlier, I can see that it's created a file here today at 11:14. I can open that up in Excel. And now just pull that up onto the right screen. So now I have the field book that I can go forward and use for my field trial. And what I like to do within this is create another tab here and call it maps. And then I go back into my R environment. And then what you could do is write these sketches out as CSVs and copy and paste them, but you can also equally just select them all from here, copy, and then go into your maps tab. And then, so alongside your field book, you also have a map of Rep 1. And then you can do the same thing. So you have a map of Rep 2. So that just allows you to, and then you can neaten it up and organize it in a way that's suitable for you. But that gives you your experimental design and a field book that you can use during your field season. One thing I also wanted to talk about is that this package has quite a nice function called zigzag. So zigzag will basically take any experimental design that you make, and it will alter it so that the plot numbers are ordered in a serpentine fashion. So you basically take your, so you create a new object called alpha lattice zigzag, and then you run that function on the design that we just made, and then you can view it like that. And then what you'll see is your plot numbers are now ordered in a serpentine fashion. So if I view, if I just go back here and view the sketch 1, you'll see now this is plot 1, 2, 3, 4, 5, 6, 7, 8. So 8 is the genotype 5. You can see that there. And then instead of plot 9 being this one, plot 9 now becomes this one, so we can check that here. Plot 9 is 228, 228, and that means when, for example, if you're in the field and you're scoring something in the field, or you're measuring in the field, you can go through like this in a serpentine fashion rather than having to go back and forth each time. So it's just quite a useful thing that you can do when you're designing your experiments. So I'll go back to my my slides now. So as I mentioned, I'll now provide a kind of couple of case studies of how we analyze data that we generate with our maize population. So this is field trials that we had last year, the maize magic population growing in Cambridge last year at NIAB. So you can find more information about this magic population by following this citation here. So firstly, we'll talk about flowering time. So we screened a number of traits relating to flowering across this population. So we screened how many days after germination it took to a particular plot took to reach tasseling, anthesis, and silking. So each plot in this instance receives one data point because we score it on a plot level rather than on some kind of plant level. So we say a particular plot could be at any one of these stages when at least 50% of the plants are tasseling or showing signs of anthesis, for example. So now I'll show you a little bit like a simple overview of how we check some of the quality of the raw data that we generate, and how we determine the heritability of these traits and extract BLUPs, as Tally was talking about earlier. So I'll open up the second part of the workshop here. I'll just clear up my environment. So the one package we need for this this part is the LME4 package, and that's the one that Tally was talking about earlier, and that's used for constructing linear mixed effect models. So I'll load that up now. I'll reset my directory, and I will read in my maize flowering time data, which is stored in a CSV in that directory, and I'll call it maize flowering data. And we'll have a look at the top 25 rows of that with this line here. So here you can see we have a data frame of 9 variables, 9 columns. So we have these ones here are all relating to the experimental design. So we have the plot ID, the identifier of the plots. which replication that plot was in, which block that plot was in, and which column it was in, and also which accession is in there. And then it has 4 trait values. So we have days till tasseling, days till anthesis, days till silking, and the interval between anthesis and silking, which is an important trait in maize because you want those to be fairly well lined up. So in terms of some initial kind of quality checking of the data, one thing that I always like to do is compare the data that I've generated from Rep 1 on with the data that's been generated in Rep 2 on an accession by accession basis. So for that, in this instance here, that would be comparing if a particular accession that flowers early in Rep 1, did it flower early in Rep 2? And that kind of is a nice check, because then you can make sure you haven't got any mix-ups with potentially in your experimental design, or one or some accessions not being what you think they are. So to do that, initially I just want to subset my data to only include the accession column, the replication column, and days till anthesis column, and then also only include in the first instance just the first replication. So I can do that with this. Then I want to rename the columns, and then I do this so that they're accession, replication, and days to anthesis Rep 1, and then I do the same thing for Rep 2 like this, and I can check what my data has looked like after that subset. So I've created 2 new data frames, days to anthesis Rep 1 and days to anthesis Rep 2, and I'll look at the heading of those. So for days to anthesis Rep 1, I've got all the accessions from Rep 1 and days to anthesis in Rep 1. Same thing for Rep 2. Then I want to merge those 2 data frames together. So I'm calling this days to anthesis Rep compare, but I only want the first and the third column. So I can merge that now, and I want to match them based on accession, so to line them up in that fashion. So now you can see we have accession ordered like that, and each accession has a days to anthesis in Rep 1 and a days to anthesis in Rep 2. And then there's a couple of plots that I want to look at to kind of do the quality checking of this data. So I arrange my plotting environment so that I can have 2 plots on one line with this line of code here. And then the first thing I'm going to do is plot a scatter plot of the days to anthesis in Rep 1 against the days to anthesis in Rep 2. So you can see it looks like we've got a pretty good agreement. I can plot a y equals x line with this line here, and you can see there seems to be a good level of agreement. I can blow this up so you can see the plots that flower early in one rep flower early in the other, and likewise this extreme late line is late in flowering or reaching the anthesis stage in both reps. Then another thing we can do to check if there is any extreme effect of the experimental design on our days to anthesis is generate a box plot of our data and separate the box plot out. So we have 2 plots for each replication. So I'll do that here, put that next to it, blow it up again, and you can see again we've got this nice level of agreement here, and it doesn't look like there's any extreme replication, and the data is relatively normally distributed across the two replicates, so that's good. So now I'm going to clear up the environment and get rid of the data that I don't need anymore, and then we're going to use the LMER function that Tally has talked about earlier to construct a linear mixed model to estimate heritability of this days to anthesis trait and extract blocks. So LMER, we can get some information about it. So this fits linear mixed effects models to data, and what it needs is a formula that you want to apply, and you need to point to where your data is. So we're going to do that here. So we're saying create an object called days to anthesis model, use the LMER function, and create a formula which is basically this. So I will then do that here. So I'm saying days to anthesis is a function of accession, replication, block nested within replication, and the data where you will find these, the data frame where you'll find these data are maize flowering data. So when you put these variables in these brackets with this 1 here, you're designating these effects as random. So we'll do that now, and you'll see we'll get thrown a warning. It did create the model, but it did throw us a warning saying boundary is singular. And you can get more information about that here. So that is suggesting, if you read this, that one of the parameters in our model is not explaining much of the variation that we're observing in days to enthesis, and that's a problem because that potentially means the model is being overfit. So we can look at the summary of the model here and see, and particularly focus on the random effects. So you can see here the random effects due to block nested within replication is very close to zero. So it's not really contributing much to our model. And that's kind of why I was saying earlier that doing this alpha lattice design and having these incomplete blocks is cost free, because if you don't have to account for them, that's fine. Which we do now in a subsequent model where we just say, because of this, for this reason, we generate the same model but don't include this term here, the block in replication. And if we run that now, you'll see that we don't get thrown that warning. So now we can look at the summary of that second model, and we can see the random effects. By far and away, most of the variance that we're observing is due to accession or genotype, but there is some error variance, and there is a minor bit of variance due to the replication in our design, which is what we saw in our plot here. It doesn't look very different, but it is subtly different. So now one thing we want to do is estimate the broad sense heritability. So that's the proportion of the variation that we observe that's due to genotypic variation as opposed to total phenotypic variation. So we can do that by extracting these variance components using this function var_core and putting it in a data frame. So we do that now, and then we've created a data frame, and then the values that we're particularly interested in are these ones. So we can say generate a ratio of the value that's in the first row of the fourth column, so that's this one with this, so that gives you 34. And generate that ratio with the sum of everything in the fourth column. So that's sum everything in the fourth column, which is that, and call that days to anthesis heritability, or H2. And then if I look at that, you can see the heritability of flowering time in this particular experiment is 0.82, which is quite a high estimate. So it's saying 82% of the variance in flowering time is due to genotypic variance, which you would expect for a trait that is under strong genetic control, like flowering time is. So the next thing we want to do is work with that model to extract the BLUPs. So these are the breeding values that Tally was talking about earlier. So that's the estimation of the random effects from the model, but the random effects that are due to genotype. So the function you can use to do that in LME4 is ranf. So you can get information mapped here. So you can see it's a generic function to extract the conditional modes of the random effects from a fitted model object. So we do that by running ranf and specifying the model that we created. And then we can see them here. So we get BLUPs for replication, but we're not interested in them. We're interested in the BLUPs and the random effects that are due to genotype. So here, everything is concentrated around 0. So if something is above 0, it will be considered having a late flowering time. And if it's below 0, it will have an earlier one. So SSA 386 has a value of 15. That would be 15 above 0, so it's quite a late flowering one. But then you can see this one here, SSA 305 is -13, so that's an early flowering line. So what we're doing with the rest of this code is we're just saying that can you just extract just the BLUPs due to accession. So that's that. And then you can see we have a data frame where the rows are the accession name, and then we have the days to anthesis BLUPs. Then we just edit the data frame a little bit to neaten it up. So we take the row names and create a new column, which is accession name, and just run through that. So then you end up with a data frame where you have 2 columns. One is your days to anthesis BLUPs, and one is your accession. So one thing you can do to make these values a little bit more relatable in terms of referring to the trait that they're actually they're associated with, you can have a look at your model and look at the fixed effects, and this value here, this estimate, that's essentially the population mean for your trait of interest. So then what you can do is you can add that value to all of your BLUPs here, which is what we're doing here. So and that is what people call sometimes a predicted mean or an adjusted mean. So what we're doing now is we're taking that data frame, we're creating a new column called data and thesis adjusted, and we're saying for this, please take this column here and add 94.985 to it. So if I do that now, go back and look at the heading, I have an extra column which is our predicted or adjusted mean for days to end thesis. So now I can look at the spread of that data with a histogram. You can see that's what our data for days to end thesis looks like. It's fairly normally distributed, but most of the variation, but maybe skewed slightly to the left. And I guess the proof in the pudding on what your data looks like can come from something like some genetic mapping or a GWAS, and we've taken this approach, we've fed this data into genome-wide association mapping, where we're trying to associate phenotypic variants with allelic variants of single nucleotide polymorphisms across the genome of maize. And we identify multiple significant SNPs, including one highly significant one here, which is right in the region of a ortholog of the flowering locus T gene in Arabidopsis, which is known to control variation in flowering time in maize. So these data are high quality, and which kind of affirms that our collection of the data and our initial analysis of the data is working well. So now on some more kind of specific photosynthesis related traits that we are interested in CAPITALISE. So in this particular example here, I'm going to talk about non-photochemical quenching and some of the data that we're generating relating to that. So non-photochemical quenching is a method of photoprotection to high sunlight to avoid, and that allows the plant to avoid cellular damage. And specifically, it's kind of the biochemical dissipation of excess excitation energy within photosynthetic apparatus, and it dissipates it so that it has heat. And you can measure it through chlorophyll fluorescence imaging. And within CAPITALISE, one of the things we're really interested in doing is measuring the kinetics of MPQ in response to dynamic irradiance. So you can see MPQ increases with when you subject a leaf to higher radiance, and then when you put it into the dark, it decreases because there's no quenching of excess energy is required. So in this specific case study, I'll illustrate some analysis of maximum MPQ in the light. So that's kind of the rate of MPQ at this point here, with the red circle is. So here, higher values of MPQ are indicative of more effective photoprotection. So in terms of the practicalities of measuring this within our CAPITALISE project, so this is the same population or the same experiment where we were measuring flowering time. So here we screened 3 samples per plot. So we took 3 leaves from a plot, brought them back to the leaf where we have our chlorophyll fluorescence equipment, and we measured the MPQ kinetics to the dynamic light. And here, so essentially it's slightly different from the flowering time data where we were generating 1 data point per plot. This time we're generating 3 data points per plot. So now I'll go through a similar thing in R that I did with the flowering time data, but for the maximum MPQ data. So I will minimize this as part of the code, and I will just clear up my environment quickly. Okay, so as before, we're working with the LME4 package. So we route to the directory of interest. We read in our maize MPQ data, call it maize MPQ data, and we'll have a look at it. So very similar to what we had before. So we have the identifiers that refer to the aspects of the experimental design. So we have the identifier of the plot, what column it's in, what block it's in, what replicate it was, and which accession is in that plot. Then, in addition, we have our 3 values for maximum MPQ, because remember we took 3 leaves. Occasionally we have NA values because potentially a leaf was damaged or lost, or in our initial, when we're doing the modeling of our MPQ data, we have cutoffs which we can use to just throw out data if it's clearly wrong. So that I won't go into now because it's would be for another workshop, but that's why we occasionally have NA values. But in addition, another thing that we have that we didn't have for flowering time is date. So date here is the day that we measured that particular plot. So this, although we're doing kind of cutting edge high throughput phenotyping, we can't measure this entire experiment in one day. So things are measured over a span of several days. An MPQ, most of the traits that we look at are incredibly environmentally sensitive, and environments fluctuate from day to day. So it's really important for us to have a record of when we measured a particular plot for a particular trait. So the first thing I want to do is similar to what I did with the flowering time. I want to compare maximum MPQ in Rep 1 with maximum MPQ in Rep 2 on an accession by accession basis. So doing the same thing as before, I'm subsetting columns 1, 5, and 10, which is this one, and oh no, sorry. The first thing I want to do is generate a value for maximum MPQ that's a single value that's of reference to the plot. So what I'm going to do is generate mean MPQ for each individual plot. So I can do that using the function row means. So what I'm saying is here is generate the row means, but only use these 3 columns, and create a new and then create a new column called plot mean max MPQ. So I can do that here. Look at the top of the table again, and you can see we have the same columns as before, but we have an additional column here which is plot mean max MPQ. So now I want to do what I was getting onto earlier of comparing this mean value across the 2 reps. So I subset the columns of interest, so it's the mean, the replicate, and the accession identifier in Rep 1 and Rep 2, and create a Rep 1 data frame and a Rep 2. Look at the heading. So now we have a data frame which is just our accession, our replication, and our max plot MPQ in Rep 1. Then merge them together, but again only take the first and the third column, and then check what our data looks like. So now we have max MPQ for each accession in both reps, and now there are 3 plots that I want to look at to kind of see if there's anything potentially wrong with our data. So I want to have 3 plots on 1 row. So the first one is the same as before, so it's plotting max MPQ Rep 1 against max MPQ Rep 2. Looks like a pretty good agreement. Again, I can plot a y equals x line to see. It looks like there might be a slight reduction in MPQ in Rep 2. If you see, most of the points seem to fall below the 1-to-1 line. And we can check that by again doing the box plot where we split things up by replication. And indeed that is the case. It looks like max MPQ is slightly reduced in Rep 2 compared to Rep 1. So this is something we're going to want to adjust for in our modeling. Then the other thing I want to do is do the same thing, but here I want to split up our data by the date that the data was generated. So I can do that by saying split it up with date instead of replication, generate that here, and then I want to blow that up and look at it. So you can see across all the dates that we measured, so there's maybe 16 or 17 dates, there's quite a bit of variation. So you can see this one here by far has the lowest average max MPQ, and it's quite different compared to this one here. So this is so this has probably one of the highest average MPQ of the day. So this is kind of harking back to what Tally was talking about earlier. You want to generate as much information as you can on how every component of your data was generated. So this is clearly something we want to account for. So we should do that in our modeling. So now I'm going to clear everything up and go through to that modeling. So now we're going to generate a model the same way we did the first time. So we're saying the plot mean max MPQ is a function of accession, replication, block nested within replication, and date. So we'll go ahead and run that model. And we can see that we're getting thrown that warning that something is, some term in our model is not really contributing much. So we can look in the summary, and again, it's block within replication. So that's not a problem. It's a cost-free thing to add to our design. So we can just get rid of it. So now we just have the three terms: accession, replication, and date. Look at the summary. So now we can see, again, it's encouraging that most of our variation is coming because of accession, but we do have a substantial amount coming from date that we definitely want to account for. And there's also a little bit coming from replication. And as always, there's some error variance. So, so one thing I didn't talk about, and Tally had talked about it a little bit, is evaluating how well your model is working. And one thing you can do is plot the residuals. And this is kind of what Tally was talking about earlier. So here what we've done is we've plotted the residuals. So this is the difference between our observed values and our modeled predicted values, and we plot that against the predicted values or the fitted values. And this allows us to visually inspect how well our model is sort of explaining our data. So ideally, you want to see that your residuals are symmetrically distributed around 0 on the y-axis, and they want to be clustered around there. And you don't want any obvious patterns in your data. If they're following some obvious trend, that means there's something that you haven't accounted for in your model. And I would say in general, in my experience, you have to take a pragmatic approach when you're evaluating how well your model is working. As long as there's no hugely wayward points or obvious patterns of distortion, you can probably proceed with relative confidence. The only thing I might say with this particular example is we get this bunch up here when we get to the higher rates of maximum MPQ. And I think that might be, if we look at our raw data in a histogram, you can see we get this tail here. So our model might not be working so well at predicting these higher rates of maximum MPQ, but again, we can go forward. And as I said before, if we get good, in our case, mapping results, and we're pointing towards genes that we expect to be involved in MPQ, then we're probably okay. So we can calculate heritability by using this var core function, as I mentioned before. So you can go ahead and estimate the broad sense heritability of maximum MPQ exactly as we did before, and we can see it's 0.59. So that's quite high for a fairly environmentally sensitive trait. So that's encouraging. We've got nice genotypic variation for the trait that we're interested in. Then going to go ahead and calculate BLUPs exactly as we did before and add the population mean to generate an adjusted mean. So in the interest of time, I'll just run through that quickly. And then there's just finally, the two plots that I wanted to show you to kind of illustrate how this is, what this modeling is bringing to us, rather than, let's just say, working on means. So the first is plotting figure margins too large. Okay, just go and plot it like this. So this is this is showing the variation, the variation in the BLUPs that are added to the population mean for maximum MPQ across the population. You can see our variation goes to about 1.9 to about 3.4, 3.3. Then I'm going to, with these lines of code here, I'm just going to quickly generate the mean values. So just do that there and then plot those in a histogram. So these are the means. So what you can see is, if we compare, I can't get it side by side, which is annoying, but what you can see here is we're shrinking the variation. So we're only focusing on the random effects that are due to genotype here, whereas this one is incorporating the variation that's due to the date that it was measured, for example, and also due to the variation that's imparted because of the experimental design. So that's that kind of shows you the power and the importance of doing these kind of analyses. And then just finally, we can plot that mean against the BLUPs added to the population mean, and you can see that the data that we generated is highly representative of the mean. But if we plot a y-x line, you can see the values aren't exactly the same, which is good, and we wouldn't expect them to be. But if we plot a linear regression where we're saying the adjusted means are a function of the true means, you can see it fits quite well. So this is a really, gives us confidence that what our modelling has been appropriate. And then just finally, as with before, I can't go into detail with it too much because it's all unpublished new work, but we're performing association mapping on this maximum MPQ, and we're identifying very strong signals, and some of these signals lie slap bang right next to the genetic position of genes that we know are involved, that are well characterized to be involved in non-photochemical quenching. And we're also identifying putative candidate genes that we have high confidence in based on gene ontology and other evidence. So I don't really know how we're doing for time, but that's everything I wanted to talk about today. I will stop sharing my screen and say thank you for your attention.
Well, thank you. That was really clear and comprehensive. As someone who's only used GenStat to analyse any data, I think even I'd have a go at having a go at it in R actually. And I thought your GWAS data looked really nice to see. You're identifying Q-sharps, I suppose, aren't they? Yeah.
We're pleased with that.
Yeah, they look really nice. Did you make sure date is not confounding with the reps or blocks?
No, we did. There's no, because, well, that could be so I think I understand the question. That could be a problem if you measured if you went through your experiment and measured all the blocks on one day, but we didn't do that. So all of our measurements, we try and time all of our measurements to co-occur with silking. So we try and do our measurements a few days after silking, because in maize and in barley we do heading, and the reason for that is because we want to measure the leaf subtending the ear when photosynthesis is likely to be mostly contributing to yield, and because everything is randomized and flowering isn't associated with the experimental design, we don't have an issue with any block or replication being confounded by date. So no. But that's an important point, and you definitely don't want to measure one block. You don't want to measure it in a systematic way in terms of block. You don't want to measure block 1 first and block 20 last and then go through like that. That would be a problem.
Did you account for edge effects using the Lattice design? That's from Peter Woolley.
No, we didn't, and we haven't done any spatial correction either, and it's something that I would maybe like to talk to Tally about doing. So no, we didn't do that.
Yeah, because when he was talking about blocking in a greenhouse, for example, when you think of the size of what, and I can understand putting a guardrail, but the thought of putting a guardrail on a glasshouse experiment, I sort of think that's going to be a massive amount of plants.
I would say one thing we do have is we do have a border around all of our experiments. So it's not like our plots that are on the edge of the design are more wind exposed, let's say, to something in the middle. Well, they will be in the nature of them being at the edge, but we do have a quite substantial border going around for our experiments.
So I've got to carry on that question. The question is, are these absorbed by the analyses? I presume the edge effects, are they absorbed by the analyses, or will you try and put them into the analyses? Any edge effects?
Yeah, I'd like to. It's just not something I have experience with, but I can hopefully speak to someone like Tally about doing that going into the future.
So were you surprised that the dates gave you different values for NPQ? Because if they're being measured in a controlled environment as opposed to in the field, should date change too much if you're putting them under a particular light regime?
Yeah, that's a good question. So NPQ is sensitive to other things other than just date. So it's sensitive to temperature and other biotic environmental stimuli. So you could imagine that one date later on, if a leaf has been, or a particular plot has been subjected to a couple of days particularly hot, that might have kind of some legacy effects on the day that you do your measurement compared to one that's had a couple of mild days before. So I guess not completely surprised, no.
Okay, I've got two more questions here. Again, it's about the edge effects. Do you look at neighbours in the design so that the more related individuals are not around each other, or is that not a problem?
We don't do that, and it's not something, well, in the magic population, everything is kind of equally related anyway. In our natural variation in our barley population, that could be something that we could consider, but again, we do it all randomly, so I think the chance is that particularly related, I assume he's talking at a genetic level, particularly related accessions are not necessarily going to be in adjacent plots, but it's not something that we put into our design.
Thank you very much, that was great. And in the interest of time, I think I'm going to say let's have a tea break now, or toilet break, whatever you want to do, it's up to you. You've got, so, and let's meet back up at 12 o'clock, okay? Well, so I think we're coming out to 12 o'clock. So welcome back, everybody. Before I welcome our final speaker, I'm just going to ask you to do another final poll, which actually is about whether you have attended any of our previous workshops. So there's a phenotyping workshop, there's a 4 genetics workshop, whether you attended both or none at all. So submit your answers just out of interest to see how popular they are, really. Okay, I'm now going to close the poll and share those results. So there's majority of you haven't attended them, and there are people that have attended both, which is nice. If you are interested, they are available online as recordings. So we'll put a link into we'll show you where you can access them later on. But they are very useful. I've listened to them and I've really enjoyed them myself if you're interested in phenotyping or the genotyping analysis. So this leads me to our final speaker, Professor Johnathon Napier from Rothamsted. I'll hand him over to you.
Well, thanks very much for the chance to tell you about our omega-3 project. You know, what's been great listening to the talks preceding is really it's emphasized, and the importance of doing field trials is something that sort of, I guess, probably everybody attending this workshop understands the importance of doing field trials. The problem and the challenge that we have is actually the many people who are not attending the workshop who don't appreciate the importance of field trials. And probably I would put myself in that category if I went back 10 years. 10 years ago, I'm a plant biotechnologist, and most of my research career was spent, you know, in the lab doing lab scale experiments and maybe getting to the point of translating stuff and growing plants in the greenhouse. And the idea of going into the field was like, you know, well, you know, that's what farmers do, and you know, why would you want to do that when you can be nice and snug and cozy in your greenhouse? And it took me a long time to realize that way of thinking is really quite prevalent in research in academia, and there's almost there's an inertia and a reluctance to try and move out of the lab and into the field. But it is the absolute missing link in the research environment, and demonstrating application and utility and value of primary discoveries. And that's really what I want to try and emphasize in my talk this afternoon. And I'm going to give you some examples from my own project, which is a GM project. But I'm not going to labor my research because I've sort of talked about it so many times now. I bore myself, let alone the audience, because we've been doing this project for a very long time. But I think there are some key messages that can come out of the project. Okay. Yeah, and this really, if you watch the little capitalized video, I mean, it chimes exactly with what you know, what my thinking is as well is that, you know, that we have global challenges. It's really obvious we've got global challenges, you know, with the climate crisis, food security. All of these things are really massive challenges and problems that we have to try and solve. And you know, the most recent challenge that's faced mankind, obviously, it was the COVID-19 pandemic. And what became clear from that was that it can be solved by science. And it is, in fact, it's only going to be science that is going to get us out of these problems. And this is the way that I see understanding and developing food security issues, climate resilience, all of these things. You know, it will be science that solves this problem for us. And you know, this is summarized by the Sustainable Development Goals, which you know, quite chasteningly were published in 2015 with the ambition of meeting some big targets by 2030. So we're halfway towards that goal of 2030, and I think, despite lots of laudable efforts and lots of focus and attention, we are still some significant way off of coming close to meeting our Sustainable Development Goals. You know, so the one that I guess you know, in plants sciences people are most familiar with are probably the Sustainable Development Goal 2, zero hunger and good nutrition. But you know, there's lots of, you know, this is not to say this is more important than any of the other goals. All 17 of them are important, but you know, they are a massive challenge for us to try and address these. And I've really sort of focused my research career in the last decade, so before these goals were published, in thinking about how to translate research. How can my work, my basic work in plant biotechnology, how can that deliver a benefit that is going to address and maybe mitigate some of the challenges that we face? How will it help towards meeting the Sustainable Development Goals? So let me tell you about my omega-3 project, which can be used as an example of how maybe you can use plant biotechnology to meet the Sustainable Development Goals. And the reason why I think it's important to flag up that this is a GM project, and although the previous two speakers were really talking about, in the main, about conventional plant breeding, which still has a massive and important role to play in meeting food security goals and many other aspects of meeting the Sustainable Development challenges. So GM and also gene editing, which I'll mention in a minute, are tools that are used extensively in academia. So, I mean, when you think about actually how many people in academia you probably use conventional plant breeding as opposed to using tools like GM and gene editing, that it is the latter, it's the GM and the gene editing tools that are probably much more prevalent than people using conventional plant breeding. So I guess what that means is that in academia, in the fundamental research base, there is a huge resource of material, discoveries, innovations, whatever, that have been developed by GM and coming in line gene editing. But a lot of that is not being translated. A lot of that is not being evaluated even. A lot of it is just produced as a proof of principle or just an experimental outcome, and is sort of left and nothing moves forward. And it really worries me that this is the case, because if we're going to try and deal with the challenges of climate change and all of these other problems we face, then we need all of the tools in the toolbox. You know, which is a cliche, but it's true. And to me, the big challenge is, how do we get more of the discoveries that we've made in fundamental research labs using GM and GE? How do we get that out of the lab and into the field? How do we get it tested? How do we get it evaluated? Because most of the time, it's not even tested. It just sits in the greenhouse or in a CE room, and people write a paper saying, oh, this could be really useful for solving food security or whatever. But actually, they don't demonstrate anything. And actually, those of us who've gone into the field and those of us who know that actually doing field experiments, what they involve, will also know that the attrition rate, the failure rate, is really, really high. You know, so things look good in the greenhouse, you go into the field, it's a totally different world. And things don't, you know, perform as well in the field as they might do in the greenhouse. And that's okay, that's just normal, but you need to find that out. And that's the thing that sort of, I start to sound quite probably irascible when I talk about it, because lots of people are quite content not to take that gauntlet up, not to take that challenge of finding out whether something works in the field or not. And I think it's vital that we do that, because probably in all the labs, you know, you think it's hundreds and hundreds, thousands of labs all over the world that are doing fundamental discovery research. Some of them, just by pure probability, must have made discoveries that could be useful for helping resolve the challenges that we face. We just need to capture those innovations. So that, I know that sounds super preachy, and I apologize, but it's my personal little sort of hobby horse. So let me tell you about my omega-3 project. So why am I doing this? I'm interested in making omega-3 fish oils in plants. Why would you want to do that? Well, omega-3 fish oils are beneficial for human health. They are proven to reduce your risk of cardiovascular disease. That's, you know, so that's a good thing. They're a limited natural resource. You know, contrary to popular belief, there are not plenty more fish in the sea. It's not the case. They are a limited natural resource, and we're harvesting them all the time in an unsustainable fashion out of the oceans. We as humans, animals, most vertebrates can't make omega-3 long-chain polyunsaturates, which is what the technical name for these omega-3 fish oils are. We can only get them from our diet, so we have to get them from our diet, but we can't do that in a sustainable way. And there is no natural plant source known of these omega-3 fatty acids. Lots of plants make omega-3 fatty acids for sure, but they're not the omega-3s that you find in fish oils. They're a different form. So that's the basis why we set out 15, 20, 25 years ago to try and engineer plants to make omega-3 fish oils. And the fact that we started 25 years ago, and I'm still talking about it, tells you it was not easy. But we think the challenge is actually to move it beyond the lab and into the field, and that's really what I want to focus on. Okay, so this is how you make an omega-3 long-chain polyunsaturated fatty acid in a GM plant. So basically, it's really simple. That's what we thought 25 years ago. You take the genes from algae, and you need to take a whole fistful of genes. So you need to take 7 genes from algae and put them into plants, because it's algae that make the omega-3 long-chain polyunsaturated fatty acids. It's not, you don't need to put fish genes into the plants, but you need to put algal genes into the plants. So you have to put a whole pathway into the plants. And we use seed-specific promoters, and we tried lots of different plant species, and we tested it in Arabidopsis and put it into canola and put it into linseed and soy and lots of other crops. Actually, we ended up putting it into camelina because camelina is a much better chassis, a much better host for the engineering that we want to do. Camelina is really easy to transform. Camelina is a Brassicaceae, so it's like a cousin of oilseed rape canola, but it's not. It doesn't cross with canola oilseed rape. It's self-pollinated, so it doesn't have any of the outcrossing problems that you see with crops, brassica crops like canola oilseed rape. It's hexaploid. It's really easy to genetically engineer. It's just and it naturally accumulates quite a lot of omega-3 quite a lot of alpha-linolenic acid, which is the precursor of omega-3 that you need to convert to the omega-3 fish oil form. So that's all great, and it works well. This is just a diagram of the construct that you need to put into the plant. So you're putting 7 genes in. It's, this is a complicated piece of genetic engineering, way past the level that most of the GM traits that are commercialized, which are things like Roundup Ready and BT insect resistance, are sort of single genes or 2 genes. This is a very complicated bit of genetic engineering, and that in itself tells you why it's probably important to do field trials. And this is just a picture of the fatty acid profile of the seeds that we've engineered in our camelina. And the thing to remember is, to appreciate, is that we're just using seed-specific promoters, so that these algal genes are only expressed in the seeds of the plant. They're not expressed in any other part of the plant. And this is the fatty acid profile of a camelina seed, wild-type camelina seed, unmodified. And this is, this is what it makes, the seed oil makes. This is what you're trying to replicate. This is what a fish oil looks like. So this is what you would get out of the ocean. So this could be, an anchovy oil or something like this. This is the thing that are harvested in the millions of tons to produce the fish oil that is used for lots of applications, but mainly fish farming. And so these are the two key fatty acids. These are EPA and DHA. These are the omega-3 long-chain polyunsaturates. These are the fatty acids that we are interested in. These are the omega-3 fish oils, if you want to call them that. That's the shorthand that we use. You can see they don't naturally occur in any vegetable, in camelina or any plant oil. There is no plant that naturally accumulates EPA and DHA. So you can only make them by genetic engineering. And this is what, this is what our camelina seed oil looks like. So basically, you don't even need to look at the numbers. It has EPA and DHA, the same levels you find in a fish oil. So, on one level, you think, okay, well, this is great. My work here is done. I've made EPA and DHA in my transgenic camelina plants. I can now retire, or I could go and do another project. And in lots of academic labs, and it sounds like I'm slacking off other academic labs, in lots of other labs, people would consider that this is that their job is done. They've demonstrated they can make a fish oil type in plants, and that's it. And this is where we sort of diverged from everybody else, because actually we think, well, actually, you know what? I think our work is just starting, because if we've got something that is as good as a fish oil, then now we need to demonstrate that actually you can use that as a substitute for fish oil, and that you could scale this up and it works in the real world, because growing it in my greenhouse is of no good whatsoever to anybody else. We need to demonstrate that it works in the real world, and that's actually, and that's sort of sounds quite sort of pompous almost, and it certainly was something that when we articulated it to ourselves 10 years ago, seemed like a good idea, but we didn't really know how to do it, because doing GM field trials in Europe is not straightforward at all, as we discovered. And yeah, so what we discovered was that doing translation of GM research is not straightforward at all. And we're lucky that at Rothamsted we had some experience of running GM field trials in the 1990s. And the problem was that those trials got vandalized by activists. And we'd also previously, in probably 2011, so the year, or 2012, the year before we started running our GM camelina trials, we'd also run a GM wheat trial at Rothamsted, which had also been threatened with vandalism. So we were sort of super sensitive about this, and also our research council, BBSRC, had actually paid quite a lot of money, in fact more money than the research project, paid to have a secure site to allow us to do our GM field trials, which is what this picture shows. So you have this rather imposing, or even intimidating field trial site, which is sort of surrounded by these large fences, and for a time we even had security guards and guard dogs and things like this surrounding the trial site. We don't have any of that anymore. But it actually, although on one level it protected the trials, it also sent a negative message that there was something, dangerous or something that needed to be contained, and that's really not the message that we wanted to say. Anyway, so this is, this really what I'm going to tell you about. The rest of the talk is about the trials and tribulations of doing GM field trials in the UK, and actually how it's actually got easier. It's got much easier. But that's only because we've learned how to do it, and also because we've spent a lot of time talking about why we're doing it. And really the key thing, I think, is explaining why you want to do it rather than how you're doing it. But I'll tell you a little bit about how you're doing it. But I always show this picture as well because it shows our farm manager at the time, Stephen Goward, going into the compound, past the security guard, and he's being videoed by someone. And if you've ever been to a demonstration or something like that, everybody's filming everybody nowadays. That seems to be the norm. But this is not the case here, actually. This is actually a BBC camera person, and she's filming us for BBC Countryfile, because we invited the BBC and media to come and film our GM field trials as part of our attempt to try and explain what it is we're doing to as many people as possible. So, rather than being sort of, trying to be covert and not tell people what we're doing, we'll tell anybody what we're doing, and anybody has always been welcome to come and see what we're doing at Rothamsted. The Rothamsted farm site is, in fact, it's quite large, and it is got lots of public footpaths through it. So there are always people walking their dog, riding their horse, out jogging through our farm site. So we try to be as open as possible about what it is we're doing and explain to them why we're doing it, so they don't think, oh, that's where that super scary GM field trial is. They just think, oh, that's just another one of Rothamsted's experiments. So doing a GM field trial, that's very different from doing a field trial, a regular field trial like the type that John Tally talked about earlier. To do a GM field trial, you have to get what's called ministerial consent, and applying for a ministerial consent takes quite some time to do that. You have to produce a dossier to apply to DEFRA to get the consent. The dossier is subject to public comment for, I think it's 30 days or maybe 48 days. So it's put on the government website. Anybody can comment on this, which means all the NGOs write in and tell you that you're a very bad person for even thinking about doing these experiments, etc., etc. But much more importantly, your application is assessed by ACRE, which is an independent committee called the Committee for the Release to the Environment, and they will look at your application and what you want to do and assess it for a whole range of parameters, including experimental design, including environmental impact, and all of these things. And they will make a recommendation to DEFRA about whether or not you should be given consent to do your field release, field trial, and then you can proceed after that. So it's not something you can do on the spur of the moment. You need to really spend several months planning your application. Then your application will be evaluated for several months, and it takes some time to do that. And so it is for research purposes only. It is not for commercialization. So we're only talking about doing relatively small scale field trials. In principle, there is no upper limit on the size of what a GM field trial could be. The biggest we've done is probably something like 4,000 square meters. But I think once you move beyond that sort of scale, then it becomes more difficult to convince ACRE, the committee, that what you're doing is really a research trial, as opposed to what could be perceived as scaling up for commercialization, which then becomes something different. So this is what I'm telling you, I guess, is that this process is highly regulated. And again, this is, I put this picture in because the first time that we did a field trial, although we're and I've made great pains to say, we're keen to move beyond the lab and into the field. The first year that we did our field trial in 2014, we ended up hand sewing our camelina. Now, we didn't do this because we wanted to do it. It's because we realized that our plot drill that we used for all our other field trials at Rothamsted wasn't actually suitable for drilling camelina. So I made myself incredibly unpopular with our farm staff, because it meant that all our farm staff had to come out and sew our trial by hand. That's not ideal. It also actually doesn't really represent what farming is like in the 21st century. at least in industrialized farming, doesn't rely on fields material being sown by hand. But anyway, we did it because it was important to do the field trial. Getting out into the field, to me, that's the key thing. Being in the environment, that's the key thing, because you want to find out, will your crop work in the real world? Because it turns out, and this is the thing that makes me laugh, is that lots of plant scientists seem to be laboring under the impression that plants grow at a constant temperature under a sort of 16 hours light, 8 hours darkness cycle, because that's what they set their CE cabinet for, or their glass house for. Of course, anybody who's ever been in the field, or actually anybody who's just, you would feel, alive, would know that actually that's not what the real world is like at all. We're conscious at this time of year that days are shortening, temperatures are dropping, all of these things, and that's just the natural cycle. But it seems to me that a lot of people don't seem to think that actually that their growth conditions in their CE cabinet represents the real world. They do not. Actually, the man in the white shirt here is the man from the ministry. That is the GM Inspectorate, and a very nice man, James Blackburn, who comes to ensure that our GM field trials are run according to the consent, to that ministerial consent. I only showed you the front page of it, but it's a long, voluminous document that lays out in very strict detail how we run our trials, what size the plots are, how many seeds we can sow, what's the sowing density, whether or not we have a pollen barrier. All of these things have to be put into the application, and then they're determined whether or not you can, how they're applied is written in the consent. And so this is what here's an aerial view of our GM field trial site in 2015. You can see our fences, which I don't really like, but they're there. And for the first couple of years, we also grew our plots under a net, which was again as a consent condition, which was to restrict the ingress of large pollinators like bees. The green section around the netted area, which is the trial, is the pollen barrier. So this is wild type camelina, which is grown there to act as a trap for any pollen. Now, this was the first couple of years that we're doing these experiments. Now we don't grow with any nets, and we don't grow with any pollen barrier, because we've demonstrated to DEFRA and to ACRE that you don't need those things. And so it allows you to progress to a more realistic representation of what agriculture would look like in a commercial setting, because no commercial farmer is growing an oilseed crop under a net or with a sort of enormous pollen barrier. That's not the case. But so that's what our trial site looks like in 2015. Actually, we've now significantly expanded it, so because the field, which is here, so this is now our GM field trial site, as well as this, as well as we also have some field trial sites over in Suffolk at Brooms Barn. So basically, I guess what I'm saying is that we've moved, expanded our field trials operations enormously at Rothamsted. So which is a good thing, because we've got more stuff to test. But in an ideal world, this is the rest of the Rothamsted farm. The Rothamsted farm is several hundred hectares. This field here is 3 hectares. So basically this is like 1% of the Rothamsted farm, but it gets a disproportionate amount of attention. And so I'm just going to show you some pictures here of as we sort of scale up. So as things got bigger, as we got more experience doing these trials, and also because of our interactions with the GM Inspectorate, who are the enforcement agency for DEFRA. So part of the story is you can build a trust and working relationship with DEFRA and with the GM Inspectorate. They're not going to let you do anything that is not defined in the consent, but you can demonstrate to them the way that you're working and the way that you're behaving, and that you've got competency to run these trials. Gives them confidence in that you know what you're doing. And so when you make your next application, then you don't need to say, for example, include the nets or have a large pollen barrier or any of these things. And all of these things just also, you know, because we said, you know, the key thing is also explaining why you're doing it, and being really open and transparent about why you're doing it. So which is why we did lots of media work in the first few years, and we were very open about why we were doing the project. In fact, much more open about why as opposed to the nuts and bolts of the genetic engineering, which isn't really very interesting to most people. You know, we're more interested in explaining why we think it's important to make a plant-based source of omega-3 fish oils, so we don't need to deplete the oceans, and that we can provide a new source of these healthy fatty acids. That to me is a much better and more interesting story than explaining about how I built a construct with 7 transgenes in it. I don't think people really are that interested. And so we do these, you know, so we talk about this stuff and have, you know, we had some very good support from our comms teams, and it allows, you know, again, there's a perception that, you know, GM is treated badly in the media, and people write about it, and you're always going to get a hard time. So probably isn't it better just to try and fly under the radar? No, it is not. It is really important to get out there and explain what you're doing. And if you tell the story clearly and truthfully, then I think you've got nothing to worry about. You know, so this is just a screenshot from some headlines from the Times about our work, you know, about our field trials. You know, I didn't describe the field trial as a triumph. You know, that's what's written in the newspaper. You know, so I think if you are careful about how you describe things, then I think you can tell a good story, and that story can be reflected in the media. And so, you know, again, some more pictures. You know, this is scaling up and scaling up. So now we're scaling up. You know, so now we're growing material in Canada. So this is 3 hectares in Manitoba. It doesn't look like 3 hectares, but it is. And you know, and you're starting to, you know, to demonstrate that the crop, you know, it grows in the real world. It grows in different environments. You know, these are the sort of things that until you've done a field trial, you think, well, you know, is that important? And like, of course it's important. You know, just because you can grow it in one environment doesn't mean you can grow it in another environment. You know, in the same way as like, just because you can grow it one year doesn't mean it'll perform the same in the next year. Those are the sort of critical experiments that I'm sure for everybody on this course is blindingly obvious. But for most plant scientists who don't do field work, it's a complete novelty. It would be a total revelation to them if somebody said, well, how many years did you do your trials for? They're like, well, what do you mean? It's, you know, and I was probably that person 10 years ago. It's like, well, you know, so it's a hard learning curve, but it's an important learning curve. And, you know, and because we did big field trials, you know, you can generate enough material, enough plants, enough seeds, enough oil to do things like animal feeding trials. So one of the main applications for fish oils out of the oceans is actually used in aquafeed, in fish farming. And so we were able to demonstrate that the oil that we produced in our plants could be fed to salmon as a replacement for fish oils that are derived from the ocean. So in other words, you know, we could demonstrate the sustainability credentials of our plant-based oil as a direct replacement for oceanically sourced fish oils. And that was a real major milestone, really important. And this is really just so this is our 2012 field trial. And this is the field trial out of Brooms Barn. So this is a nice drone shot from our visual communications team. And you can see, you know, we've got some big blocks here that are so these are the much bigger blocks where we're growing at scale, and probably these are to be used in aquafeed trials. And then this is a randomized block design, so of the type that we've been talking about earlier today, where we're sort of, you know, we're growing under different treatments, and the design has been through the Rothamsted statisticians. They've designed the randomized block, and you know, so that it is such that it allows us to derive, and it's appropriately replicated as well. So all the things that you just heard mentioned in the previous 2 talks as being really critical, you know, we're doing that here. Now we're lucky at Rothamsted that we have a really great statistics team, and we also have lots of experience of running field trials. So it allows us to do these things. You know, these sort of things are normal. In fact, you can't get into the field without having been through what's called the Field and Farm Evaluation Committee, Experimental Committee, and they will actually check whether or not what you're doing is suitable. But again, to me that was really helpful to be able to do that. And in the last few minutes, I'm just going to mention gene editing as well, because I think gene editing is something that that has, gets a huge amount of media. Obviously, last year's Nobel Prize winners were as a consequence of the discovery of CRISPR-Cas9. It's a tool that is viewed as having great potential, especially in plant breeding. And I think lots of plant breeding companies are very interested in the potential of gene editing to at least compress the breeding cycle and to allow things to be developed maybe a bit more quickly. The problem is that in the EU, there was a court case ruling from the European Court of Justice in 2018 that basically ruled that gene editing was the same thing as GM, which effectively blocked the use of gene editing in the EU. And I think, our view is, and the view of DEFRA's and the British government is that, gene editing is not the same thing as GM, because GM is effectively about adding foreign gene, extra DNA to a plant or an organism, and gene editing is about removing DNA. So the two are quite different, but the EU doesn't see it like that. Now we did in fact the first European field trial of gene edited crops, of camelina crops, and actually the trial was running while the European Court of Justice judgment came out, and somehow it basically meant that our gene edited crops suddenly magically became GM overnight, which was a bit disconcerting, but fortunately we'd sown them within our GM compound, so it allowed the trial to proceed without any hindrance. But the reason I show you this is not anything really to do with the legal aspect of it, is that this is our gene edited crop. This rather sad looking little stubby looking sort of camelina in the middle of here in this plot here, but you can see it from the drone shot here. It's the one in the middle here, and in the field this looked more like Arabidopsis than it looked like camelina. It looked really unhappy, and actually when we grew it a second year, it didn't even grow at all. It completely failed. In this year, you can see actually we were extensively irrigating our camelina because it was incredibly dry. This was 2018. You can see how dry the rest of the fields, either in the GM compound or outside, are, and we've had to irrigate really heavily our GM plots. Now, why am I telling you this? What's really interesting is that when these plants were growing in the greenhouse, they looked just like the wild type. They looked just like the parental lines. So it was only when they went into the field that they looked like they performed really badly. So if you just finished your experiment and said, well, I've made these plants, they all grow pretty well in the greenhouse, that's my work here is done, we don't need to do this anymore, or we can, we could, pass this on to somebody else, that would have been a big mistake, because these plants, and they were edited to try and make them so they had the seed oil content would have been like olive oil, so to make monounsaturated fatty acids, so something potentially useful, but in the field they just failed. But you don't know that until you've done a field trial. So to me, this is really this is the critical thing about doing field trials, and it's going to get easier. So the problem with the European Court of Justice was obviously, and that's still a problem for our partners in the EU. They still cannot do they cannot do gene edited field trials. But currently, at the moment in the UK, although the legislation only applies to England, moving through the House of Commons is a piece of legislation called the Genetic Technology Precision Breeding Bill. It's been through several stages. I think it says here it's been through its committee stage, and it's expected to come back to Parliament and to the Commons sometime this month in October. And it will completely revolutionise the way that gene editing is treated and is regulated. So I see that as a really good thing, and I think it's really exciting for anybody in plant breeding who's interested in using gene editing CRISPR-Cas9, because I think it's going to become much easier to actually see that all the way through to commercialization. It's already easier to do gene edited field trials. We did them this summer, and they could be grown just anywhere on the Rothamsted farm, and that was really exciting to do that. And so I'm just going to wrap up. And so why is all of this important? And the one thing that's probably come through in my talk, talking about my experience, is just how incredibly long it takes to develop things. And this is the challenge in general in agriculture, because you only get one, you only have one season a year, at least at the same location. Time is not on your side. And the average time for an agricultural innovation, so to go from an idea, from a discovery through to an actual product thing in the field, something that you can buy, on average is just under 30 years. In some cases, it could be much longer. So things like hybrid corn, which was discovered 60 years before hybrid corn actually became a thing. 60 years, so like an entire lifetime. So time is not on your side. So you really need to push as hard as you can. And this is why I'm so passionate about trying to explain to colleagues, especially basic research colleagues, about the importance of getting into the field as quickly as possible. It could be that your experiment in the field is going to fail. It'll be like the gene-edited camelina. It goes in the field, looks like a disaster. That's okay. It just tells you that you need to focus on something else. Because we need solutions to the global challenges that we face. We really do need help. And collectively, mankind needs. We're going to need all the help from innovation and science that we can get, and time is not on your side. So, quick summary. Probably, the key thing is here: why does it take so long for basic research to be translated? And I think the answer to that is really because we don't actually value translating basic research. We do lots of stuff. I mean, I don't. If you, as I said, there's hundreds and hundreds of research labs in the UK, all beavering away doing discovery research. How many of those are making discoveries that are being translated? I don't think there's a very big number. How many people are doing GM field trials? 2 or 3 organisations: ourselves, John Innes, Sainsbury Laboratory, NIAB. That's it. How many people are growing plants under the qualifying higher plant rule that allows you to grow gene-edited plants as non-GM? We were the first, and there were 2 other approvals, another one from Rothamsted and one from the John Innes. I'm preaching to the converted here, but people need to get into the field. I just don't understand why they don't want to do it. And the key question, just because this is my last slide, does not mean this is the least important thing. And it's really important for gene editing and GM, the concept of social license and consumer acceptance. It's always a massive issue, and people need to understand, need to be given the chance to understand what it is you're doing, why you're doing it, why are you doing it, who's paying for this and who's benefiting from it. If you're at a publicly funded research institute, I think you're in a much better position than if you work for a big company, because people are just genuinely suspicious of big companies these days. And I think that's a challenge, because for lots of other types of technology, they're quite happy with big companies providing them technology. They're quite happy with their smartphone, and they're quite happy with supermarkets. When it comes to plant breeding and biotechnology, then they're deeply suspicious for reasons that are historical. Okay, and here endeth the lesson. I think that's pretty much me finished. There's lots of people on the side there who I acknowledge who've done all the work. I just take the credit and talk for it. And that's it. Thank you very much for your attention.
Thank you very much, Jonathan. As always, put your questions in the question box or put your hand up and we will try and get you to ask a question. I completely agree with you. I think translation is the elephant in the room in some ways. I think there's a lot of people with bags of seed under their desk that they're saying, oh, this was great or that was great. But I suppose one thing would be, is there enough funding there to encourage people to translate that research, or are people always wanting to fund something new, to want to fund the next exciting new idea, which if you're not going to build on the stuff that's worked previously, then you've just got a lot of new ideas that go nowhere, haven't you, really?
I mean, I think all of that's true. I mean, I think there's probably lots of interesting material up and down the land in university labs that could go into the field, and people literally don't know how to do it. You know, what I think is really weird, if you're sort of an old timer like me, you know, can think back to the way it was in the 1990s. You know, several universities did GM field trials. You know, the University of Leeds did GM field trials. The University of Reading did GM field trials. You know, I don't think, well, in some cases, those universities have probably sold, you know, they closed their agricultural sciences department and sold their field trial sites off for housing anyway, so they probably couldn't do it. But, you know, there's sort of like an inherent loss of capability. I think that, you know, that's one issue. But the point you made about, you know, always chasing something new, I think is also a big problem. You know, we've really had to fight really hard to be able to try and continue to get funding that allows us to build on the success that we've proven. Because people always think, they always say, well, why are you still doing the same thing? Why don't you do something new? It's like, well, because this works, and I think it's important to translate it. That's why. I could go away and do something new, but then I'm just starting again from scratch. And what's the point of that? You know, meanwhile, as Greta Thunberg says, our world is on fire. And, you know, so it sort of seems to me, you know, ridiculously self-indulgent just to say, well, I'm interested in understanding how gene X regulates process Y, and I'm just going to do that. It's like, well, why don't you just do something useful? That sounds really preachy, sorry.
I think as an academic as well, if your value is based on how many publications you've done, then sometimes working on something new is going to increase your value, I suppose. So I can see that would be a potential reason, but yes, it's not the right reason.
No, I've just stopped sharing my screen, so I don't know whether that's a good thing to do or not.
What was I going to say? Oh, so given that this has taken you 20 plus years then to bring from idea to presumably a crop that you can translate, do you think now that GM has been legalized, or whatever the terminology is, do you think that timescale potentially can be shortened for crop development?
I mean, probably a little bit. I mean, the thing that I realized though is that, you know, the bit that you can't shorten is the field bit, you know, and, you know, that's still going to take you, you know, it'll probably take you the guts of a decade to sort of do the, I mean, say, for example, if you want to get full regulatory approval to commercially grow a GM crop, and I'm using the example of how it would be done in the US because that's the best, you know, the best proven pathway. You know, you're still going to have to do several years of pre-regulatory field trials to demonstrate, to convince yourself that you've got the best event, the best line that's going to work before you build, you know, before you start getting the data for your dossier. And your dossier is going to need at least 2 years field trial data, probably and multi-location data. You know, so probably collectively that's, you know, that's 5 years worth of work to do that. You know, if you can compress everything in front of that, you know, everything that's upstream of that, so you compress the discovery work, the validation, you know, some of that will be easier, but you know, it's still tough, and and actually, I mean, what's really weird for the UK is because we've left EU, and the EU rules on GM at least were effectively a moratorium. You know, we're blocking any commercial GM ever being approved. So we've obviously diverged away from that. But actually, we don't know what the rules will be because the rule book has not yet been written about how would you go about commercialize. You know, what are the rules for commercializing a GM crop in the UK? We don't know. You know, so that in itself is a barrier. I mean, so the stuff that's going through Parliament now is just specific for gene edited crops, you know, and that's good and that's a step. But actually, you know, I'm biased. I think you can do much more with GM than you can do with gene editing. So, you know, when are we going to find out what the rules are for GM?
Okay. Is there any further questions for Jonathan? Type them in now. Put your hand up. And if not, I'm going to ask if there's any further questions for the rest of the team, for Tally or for John. I have a comment here from Peter Wally. Unfortunately, many blue sky lab-based scientists either can't do or don't consider fieldwork or lack facilities, which I suppose, yeah, if you haven't got the facilities, it's not what you can do about it, is there? Unless you...
Well, I think, you know, that's where you have to sort of think about, you know, collaboration. I mean, so, you know, our gene edited field trial that I showed, you know, that was, you know, material that was provided, you know, that was generated in INRA Versailles, you know, and our colleagues in INRA Versailles couldn't do a field trial in France because they're not allowed to. So they asked if we could do it. And we've done, you know, we've also done field trials for colleagues from the US as well, in the same way as we've done field trials in the US. So I think you just, you know, what would be great is if we sort of established, you know, it doesn't have to be a formal network, it could be an informal network of people saying, okay, well, if I wanted to do a field trial, I know that Rothamsted has got, you know, 300 hectares, you know, or 20 hectares of GM field trial site. So, you know, I can go there. I worry that people just use as a convenient excuse, oh, well, I haven't got a field trial site, so I won't, you know, that's my get out of jail trial card. I don't have to do a field trial. Because in the back of their minds, maybe they're worried that it won't work. You know, that sounds terribly cynical, but I mean, you know, there's lots of people that, you know, probably could do trials. And actually, the thing that makes me laugh even more now is, of course, you know, somebody said, well, it's so expensive to run a field trial. It's like, have you seen the electricity bill you'll get for growing stuff in your greenhouse? Because it's probably 10 times more expensive than running a field trial.
Of course, now from the other two talks, we know it is technically it's a little bit more difficult as well, because you can see all the considerations you have to take into account as well. So I can see why moving from a small lab-based project, it can be tricky.
I think you just have to start somewhere, and I think, you know, and I think it's been great with the two talks preceding mine, you know, talked about really, you know, doing robust, you know, experimental design that is going to give you, you know, data that might be useful for, you know, GWAS type experiments or whatever. But, you know, you can start off by doing, you know, and we started off by doing what was effectively observation plots, where you're just putting stuff out into the field to see whether it lived or died. You know, to see how did it, you know, how could it survive an English summer? You know, which is sort of like, sounds like a fatuous question, but is really, if you can't do that, then you're sort of, then the whole thing's dead in the water anyway. So you don't need to do, you know, you could do that within, you know, you could do 20 square meters would tell you that.
There's a question here, is there any way we can encourage people to send their marvellous lab plans off for field testing when it can potentially undermine their success that they've seen in the lab? Which is a fair point, but I think you maybe have to take it on the chin. It says, you know, it works in the lab, doesn't work in the field. It's good to know.
Absolutely. I mean, isn't that, you know, that's the whole, isn't that like, you know, I don't know whether it's Google or some, you know, big tech company, you know, it's like, you know, fail fast is the mantra. You know, if, you know, isn't it better to find out that your innovation doesn't work? You find that out quickly, and then you can iterate or move to something else. I mean, to me, that's the key thing. It's okay to fail. You know, we've had stuff fail in the field. That's okay. I mean, our GM wheat trial that we ran, which was, you know, trying to engineer plants to make them, you know, repellent to insects because we'd engineered an aphid alarm pheromone, worked brilliantly in the lab, worked brilliantly in a field simulator in the grass house, went into the field, didn't work. And that's okay.
Yeah, sometimes you just need to know the truth.
Yeah,
okay.
and that's, you know, that's exactly right.
I just got a comment here. Unfortunately, the act of pursuing collaborative relationships like that, or even thinking that, may not appear on the radar depending on the research environment the scientists have grown up in. So that's just to add to that comment. So as you say, we know field trials aren't easy, but it is the way forward, and especially towards translation. I've got one further question actually. How long is it before we might be able to see your plant-based fish oils in the shops or on the shelf? Is that available?
Well, I mean, yeah, my slightly sort of tongue-in-cheek comment is, well, you know, I'll tell you what I've been saying for the last 10 years, which is in 5 years' time. But actually, I think we're at a point now. You know, I would hope that we would start moving towards regulatory approval process in the US in 1 or 2 years' time. So, you know, it's not going to be next year, but hopefully, in 3 years or 5 years' time, we would be able to actually have a commercial product, which would be great, and then I can retire.
Okay, so with that, I think we're gonna have to wind up our workshop now. I just want to say a very big thank you to our speakers for 3 really enjoyable talks and discussions, and obviously to the audience for providing this discussion with us, and to Christina Olsen, who behind the scenes has been organising the webinars with myself. If you're interested in attending any of the other workshops that we have on the forward genetics or on the phenotyping, you can find these if you follow this link in the website, on the CAPITALISE website, and you can watch them at your leisure, stop them when you need to. If you're interested in upcoming events from the CAPITALISE project, you can also sign up for our newsletter, and that has all the things we're up to in there, so we hope you can do that. Just leaves me to say goodbye from me and goodbye from our speakers, and thank you for attending, and thank you very much to the speakers. Thank you and goodbye.